Hey, real quick before we get into the actual podcast, I turned off the ads for the mid-rolls, so you should see no ads throughout the podcast. Also, there are chapters for each section, so you can skip to whatever section that you're most interested in. And you can also listen to this podcast on your favorite podcast platform, so you don't have to sit here and watch the video necessarily. And finally, if you like this kind of podcast, I have a podcast that releases monthly for the Physionic Insiders. It goes far more into the details and how to actually apply a lot of this information to your life. So if you like this kind of work and you want to support my work as well as get huge benefit through the actual monthly research, then consider joining the Physionic Insiders. It's in the description box of wherever you're listening to this. Okay, with that ad out of the way, let's get into the podcast. Hi, hello. Welcome to the Physionic Podcast. It's been, 3-4 months since the last one, and today we're going to get into yet another gargantuan podcast episode where we're going to be covering, let me rattle off the list of things we'll be covering here. Astaxanthin and skin. Dr. Rhonda Patrick on her opinion on mixing banana with blueberries, along with your comments on that. Ergothioneine and mitochondria. Glycine and longevity. I might also touch on glycine and sleep because I didn't actually have that on my list, but I forgot that I did do an investigation on that as well. Cocoa benefit for cardiovascular disease, glucose spikes causing cardiovascular disease, multivitamins for cognition, red light therapy on skin aging and mitochondria. Zeaxanthin and lutein for eye health, omega-3s for cardiovascular disease, creatine on your bones and muscle, specifically for people who are of an older age, let's say over the age of 60. Reversing Antibody therapy, which is a really cool science that I'll be getting into. 2 age-related issues that come about roughly right around the age of 60, so I'm going to use that as the benchmark. 2 things that are specific to people in their 60s and beyond that you should be aware of. And finally, C15, the Fatty 15 supplement company, and then I'll be going into a little bit of housekeeping as well, assuming that you're going to hang out with me for that time. I guess first things first, if you're watching the podcast, you'll notice the old couch is gone. I did end up selling that. I've moved since. Well, I was going to add this in my housekeeping section. Maybe I'll talk about a little bit more during that section, but I finished my PhD. I finally have my doctorate, and I've moved. So, I got rid of that old couch, and now sitting on this new couch and in a new location, which has been quite an adjustment because, especially for the videos, getting the audio and the lighting correct has been an absolute nightmare in this new place. So making incremental improvements over time. I also don't have a stand to hold this microphone, so I'm going to be holding the microphone for the next several hours. And speaking of the length of this podcast, let's see. The last episode was 5 hours long. I don't think that this episode is going to be that long. I don't know if I have the energy to do 5 hours this time around, but it will be quite long regardless. So probably if you take last episode and combine it with this episode, you'll get a full night's rest. You'll get about 8 hours of sleep. So that's what we'll aim for in total. Okay, without further ado, let's go ahead and jump into this because I don't want people to feel like I'm rambling about every little thing when we should be getting into the science. I guess one thing before I jump into this, I have videos on almost all these topics, and I'll be fleshing out some of the additional nuances in this podcast. I usually get comments from people saying, hey, you should break this podcast up into little bits. The answer is no. I refuse. Don't even comment it. It's not going to happen. But I do have videos on each one of these topics. So if you'd like to, have a visual representation, much shorter videos and whatnot, then jump on over to those. But if you'd like to relax and just sit on the couch with me and just discuss science, then this is what this podcast is all about. All right, so let's flesh out this astaxanthin and skin. So I've been the last year or so, I'd say, probably the last year, maybe a little over a year, I've been really focused on a lot of anti-aging skin supplements, molecules, things of that nature. And certainly for selfish reasons, I definitely like to take or just do things that are going to maximize my looks anti-aging wise. Not saying I'm an attractive human being or anything. I'm literally just saying that I'd like to stave off aging as much as possible. And certainly what's within is certainly incredibly important. You could easily argue it's more important, but I'm also taking care of that as well. But also on the exterior, that's something that people have to constantly consider, considering that everybody else is seeing the external version of themselves. So I looked over collagen, hyaluronic acid. Those are the big two that I covered in the past. And now I was focusing a little bit of my attention on astaxanthin just because I was curious if that might have an effect. And if I remember correctly, I went over a meta-analysis that looked at astaxanthin consumption or supplementation and the effect that it had on 3 different outcomes of skin health, skin youthfulness, whatever you want to call it. So 3 different skin metrics. It was elasticity, moisture, and the wrinkling. So those were the 3. And the mechanism by which astaxanthin does these things or is supposedly supposed to help with anti-aging of skin is that it is a potent antioxidant. And I often feel like a lot of these molecules are being talked about as just this just like they're an antioxidant, therefore they're beneficial. And it seems to me like it's almost getting overly repetitive when I think that there's a lot more that these molecules could be doing. And while that is the primary mechanism of astaxanthin to be an antioxidant, meaning that as your cells create these damaging molecules called free radicals, I'm sure we'll talk about that in the future of this podcast. But these free radicals are produced by our body. As a matter of fact, I know of one mechanism that we will be discussing called NADPH oxidase. So anyway, these free radicals get produced like reactive oxygen species, and they can be, if overabundant, damaging to your cells because they interact with a number of different proteins and fats and sugars that are found within your cells, and they essentially damage the integrity of the structure of the entire cell. So antioxidants are around to quell, think of like a reactive oxygen species as being a rebellion, and the antioxidants are there to quell that rebellion and to create it, go back down to a more normal state, a more steady state. The reason why I say steady state is because, and I've said this many times before, that reactive oxygen species do serve a purpose in our cells. It's not that we want to eliminate them entirely. We just want them to be at minimal levels until they're necessary to be used and as signaling molecules. And then they can be increased for a little bit, but then they have to be dampened back down to a baseline level. That's a really rudimentary understanding of that concept. So, but with aging, we have this continuous buildup of reactive oxygen species, as in our oxidative stress, the level of creation of these damaging molecules versus the amount of antioxidants that we have available becomes unbalanced, so that the reactive oxygen species rise and the antioxidants decrease. And what I mean by antioxidants in our cells is that our cells also have the capability of generating antioxidant proteins. Okay, so that's a little bit of a background on antioxidants and reactive oxygen species, but astaxanthin is a molecule that's unique in that its structure, it's a very kind of linear structure with these rings on either end. And those rings actually have a potent ability to quench these free radicals or these damaging molecules. And that's the primary mechanism. But through that, because it has that ability, it also decreases the amount of cytokines that are released. Cytokines are these, can be, I should state that, can be these pro-inflammatory proteins that are secreted out of the cells, which then recruit immune cells to that region. Now, that's typically a quote unquote bad thing if you're not sick, for example, because if that continuously happens over time, then we're talking about chronic inflammation. And the reason why immune cells being recruited to an area is a negative without infection present is that if these cytokines end up activating the immune cells, then these immune cells shift from, I forgot which one it is, M1 or M2. They go from a quiescent, kind of restful state, quote unquote, to a more activated state. And it's more like a gradation scale. It's not really like a flip. I think we initially thought that it was a flip, just go from quiescent to rage mode. No, I think it's a little more of a gradation scale. But the point being that these cytokines get secreted from the cells, let's say from your keratinocytes or from any number of different dermal cells that you have, so skin cells, and those can then recruit these immune cells to that region. If these immune cells are then activated, they themselves will also start to produce a lot more of these reactive oxygen species and will start to secrete more of these pro-inflammatory molecules, which ultimately, there's so many different mechanisms by which this negatively can affect your cells. And I'm talking about it from the perspective of just your skin, but it also applies to the rest of your body. Anyway, if you can quench these reactive oxygen species, you will also have an effect at the amount of cytokines that are produced, because there's a strong relationship between reactive oxygen species generation or free radical damage and the increase in gene expression of these cytokines. And I have to specify that I'm talking about pro-inflammatory cytokines because there are cytokines that are also known as anti-inflammatory. Interleukin 10 is one example of that. While pro-inflammatory, or what tend to be pro-inflammatory, keep in mind when I'm saying like pro-inflammatory, I'm saying like, let's say 90% of the time it's that way. There's always exceptions, but the pro-inflammatory ones would be like tumor necrosis factor alpha or interleukin 1 beta, those kinds of cytokines. And those are the ones that we're talking about in this situation when they're overabundant and recruit the immune cells. Okay, so astaxanthin quenches reactive oxygen species, reduces cytokine release, and if I remember correctly, let me check my notes here, it also increases the internal antioxidant systems within your cells. So the cells themselves, that's quite a word, that's quite a jumble of words. The cells themselves have these antioxidant systems that I mentioned, and they can produce these proteins that have the ability to quench these different reactive oxygen species if necessary. And some of them are pretty potent. So like superoxide dismutase, glutathione is a really popular one in the social media sphere. People say that one repeatedly. Or catalase is another one, which is found in, well, it's found in the cytosol of the cell, but it's primarily attributed to an organelle known as the peroxisome. Anyway, these antioxidants are around for at least one of their purposes, is one of their primary purposes is to quench these potentially damaging molecules, and to make sure that they don't become overabundant. So astaxanthin seems to encourage the gene reading or gene expression of these proteins that are responsible for the innate inner workings of the cell to be able to quench these molecules. One more thing as well is that it reduces the peroxidation of fats, so lipids, that are used in the production of sebum. So you know that there's a secretion that comes out of I shouldn't say pores, but this gland that's found in your skin called the sebaceous gland. And it's actually a perfectly natural process, a normal part of your body's physiology. It's probably, I think a dermatologist would have to correct me if I'm wrong on this, but I'm 70% sure that it's the reason why we get oily skin. So some people have an overly active sebaceous gland, and this sebaceous gland creates this oil and ends up secreting it. And it's also found in the follicles. Now, that sebaceous, or that sebum that gets produced, is produced at least partly with different fats. And if those fats are oxidized, then the sebum is less functional, essentially, which means that then you could you can have more acne and things like that. You can clog your follicles. So, and this is just like one mechanism. I'm sure there are many others that somebody who's like a skin pathology or skin scientist could probably talk about this in far more depth. At any rate, astaxanthin prevents the peroxidation, which is basically the oxidation or the damaging of, in this case, damaging of these fats, which ultimately leads to lower quality sebum, which then can affect the dryness of your skin and can also affect the quality of your skin as a whole. So those are the mechanisms, but does astaxanthin actually improve? And that's something I'm going to try to do throughout this whole thing, is try to discuss the mechanisms. I think there's maybe one topic where I don't have a mechanism section planned out, but I'm going to cover the mechanisms, which is what hopefully makes Physionic unique from other, hopefully everyone else. But I'm going to cover the mechanisms, then I'm going to go into the actual results themselves. So go from mechanisms to the clinical evidence, and then I'm going to go into a few comments from people that have tried some of this stuff. And some of the comments were really insightful, which is stuff that, I'm only one person. I only have a certain amount of experience. So it's great to have other people's experiences that I can collate together and report on in these podcasts. Okay, so what were the clinical results? Ultimately, astaxanthin was shown to improve skin moisture as well as skin elasticity, but did not have an effect on skin wrinkling. Now, I don't think we have enough data to be 100% certain that there's no effect on like wrinkle depth or wrinkle amount from astaxanthin consumption. So I think I would hold off on the overall interpretation of that yet. I think I would say currently we don't have any evidence that it seems to help with reducing wrinkles, but that could change if we get more data in the future, because I think the p-value is relatively low, and it wasn't like they added a ton of studies. It's not like they had a ton of studies looking at wrinkle volume or wrinkle amount with astaxanthin. So we'll see what the future brings, but so I guess what I would finish with is just to say that astaxanthin has some evidence that it provides some help to the skin. It's just not as powerful, I would say, as some other ingredients, some other molecules like collagen peptides is something that I think has gotten more and more research behind it. indicating that it does seem to help, especially with like wrinkles, but it also may help with like elasticity as well. So I think collagen peptides hits a lot more of these mechanisms, or a lot more of these outcomes, I should say, because the mechanisms are different between collagen peptides and astaxanthin. And so if you had to pick between the two, I would pick collagen peptides. Anyway, the comments. So these aren't like usually people's names. This is what I'm able to pull off of YouTube, and it gives you just their handle. So you're going to hear some interesting names here. Stacked Crooked had this to say on astaxanthin. This is totally anecdotic. I don't think that's a word. Anecdotal and worthless as evidence. I appreciate the talking about it as if, well, I'm just going to throw this out there. But I do appreciate the fact that people do understand that if you have an experience, it is an N of one. And I know that scientists say that, and people sometimes just kind of repeat it, but I don't know if they really internalize exactly what that means. An N of one can be confounded by like a trillion different things, which is not the case with studies, which is why we focus on studies. Anyway, I won't go into it right now. Back to Stacked Crooked. But I recently stopped needing reading glasses after taking astaxanthin for about 3 months. I started astaxanthin in the hopes that it would protect my skin and stop further deterioration of my eyesight. Then I recently realized that I unconsciously stopped using reading glasses about 3 weeks ago. So it seems to have not only prevented further deterioration, but actually reversed some of the damage. I didn't expect that at all. I hope it lasts. Yeah, I hope so too. And although that didn't have anything to do with skin, and it didn't seem like there were that many people that were that commented that were actually using it for skin, so I couldn't include any of those comments. But this is a bit of foreshadowing to in the future of this podcast, where we're going to go into some of the sister compounds of astaxanthin and the effect that it has on the eyes. All right, moving on to Dr. Rhonda Patrick and her opinions on mixing bananas with blueberries. So the controversy here was that Dr. Patrick was saying that you should not mix bananas with blueberries if you do, let's say if you consume them in combination, or if you use them in like a smoothie, for example. And the reason for that is because blueberries are full of polyphenols, which we know through multiple mechanisms, clinical evidence, and more, that polyphenols tend to be a net positive for our health through a number of different means. And we're actually going to go into a particular type in a little while as well. But the point being that you want to have polyphenols, blueberries have them. And unfortunately, when you mix blueberries with bananas, suddenly you lose all the polyphenols that are found in blueberries. So the reason why is, according to the study that she cites, is that the bananas have this enzyme in them called polyphenol oxidase. This polyphenol oxidase oxidizes or changes the chemical structure of the polyphenols, essentially rendering them, I guess you could say useless, less functional. And so the idea is that you should stop eating bananas with blueberries. Now, a lot of people got really upset with that, which is such a hyper-specific scenario. But let's say a person was doing that. And to be fair, there were a few people that commented and said, wow, I do that. I do combine the two. So it's just something to be aware of. If you care about polyphenols, if you don't care about polyphenols, then just forget about it. Don't worry about it. There's no problems with combining the two. But if you do care about polyphenols, it might be worthwhile to just separate the two. This is not to say that bananas are bad for you. They're not. It's not to say that blueberries are somehow useless. because they've been combined. They're not. But if you want the best bang for your buck from, a nutritional standpoint, then simply separating the two is your best bet. It's really not, it's not any deeper than that, really. Let's see. Yeah, that's basically it. I mean, that was this one study just tried to find out about that. And there are other foods, like other people commented that there were other foods that also have high levels of this polyphenol oxidase. It's actually not really the case. Bananas are among the highest, and maybe there's like one or two other foods. I think one of them is like beet something. I forgot exactly what it was. It doesn't matter. The point being that there honestly are not that many foods that actually have high levels of this polyphenol oxidase, at least from the list of like 30 or 20 that the study mentioned. Bananas were way, way up there. So I think it's really just a hyper-specific scenario that I think most people don't need to worry about. And like I said, if you don't care about polyphenols, just chuck this information. Okay, so the comments for this, I did get a few good ones. Inner Light Dimension said, if eating bananas is wrong, I don't want to be right. I understand. Please continue eating bananas if you feel that strongly about it. Miami Man 196 had this to say, every edible item has a study that says it's bad for you and another study that says it's good for you. Bam. Well, you know what, Miami Man? That is not that untrue. It does seem that that's the case, and I think you could probably make an easy argument any which way. So it's true. If you were to pick a food and then just look for studies against it and look for studies for it, you could probably collate a group of studies on both sides. The problem is that science really isn't like that. Science is a lot more fine-tuned in terms of how we talk about our conclusions. And this is something that I really stress for people to try to learn how to do and understand, to be a lot more nuanced. I've said this before in the podcast, and I've said this in many other videos before, that nuance is king when it comes to understanding how to interpret science. You're going to have some studies that have these like overall, they just look at so many different metrics, and you can get a pretty good idea of what a particular food might do. But even huge studies, unless they look at so many different populations of people or samples of the populations of people, it's going to be extremely limited. And even in my interpretations, even I bend the rules a little bit in just kind of generally saying things, when in true scientific fashion, I should be a lot more specific. The reason why I don't get overly specific is because most people just tune out the specificity. So I realize I'm being a little bit wishy-washy in my explanation here. So to give you an example, let's just use the same example we've been using, bananas. Let's say you look up a study in banana for the consumption of bananas and is it healthy? Which is completely the wrong kind of question to ask, but this is typically how people write or ask questions. So let's just roll with it. So you find one study that says bananas are amazing. They're really fantastic for, let's say, cardiovascular health. They're just great at lowering your ApoB levels. I don't know. I'm just throwing something out there. This is not fact, by the way. I'm just throwing out an example. And on the other hand, you find another study that says, well, okay, but they increase blood sugar and they increase insulin resistance as a whole. Okay, so now you found two studies that contradict one another. But the truth is they don't actually contradict one another. The thing is that now all I've done is layer on one more layer of specificity, which is one study looked at bananas on cardiovascular disease, and the other study looked at diabetes risk or insulin resistance. So while those two are closely tied together, because obviously diabetes contributes to cardiovascular disease, and people that have diabetes tend to have a higher risk of cardiovascular disease, we really have to think of it like a bowling ball. You're asking a question, and the bowling ball is this like probing at the answer, and the pins are this is completely coming off the top of my head. The pins are the answers, and you're hitting all the pins, but what people do is that they jump from like lane to lane to lane almost, and they don't stay within the confines of the lane. It's like you have to have the bumper ball, the bowling ball bumper plates have to be up, or railings have to be up, to make sure that you're always stuck to that exact definition of what the conclusion is. So in our example here, bananas on cardiovascular disease, and I teach this in my course, this is kind of one of the main things that I talk about, is being hyper specific about the questions that you're asking. So looking at bananas on cardiovascular disease, that is some level of specificity, but then where I don't go as in depth when I should, to be fair, is that you also have to keep in mind, okay, well, what population are you sampling? Okay, bananas are beneficial against cardiovascular disease in people that are in their 30s that are also very active and use the glucose taken up from the bananas. And they're very active in a very particular way. They do a lot of endurance exercise. It's like suddenly you get into these nuances. Like each study is telling you a very specific story. And unfortunately, the way that it gets reported in social media, and where I am somewhat culpable, although I do try to like offer some level of nuance, is that people will just look at the study and say, well, then it applies to everyone. Or bananas are good on this day and then good and then bad on another day, when they're looking at two different outcomes, or they're looking at different populations, or they're looking at different times, or they're looking at different geographic areas. Like it's just so many different little stories that each study tells you. And then you need to group those together, meta-analysis, to determine where there's consistency in the effects, to be able to tell, oh, okay, yes, this truly does help in this group of people in this particular metric. Hopefully that made some sense. I feel like the bowling ball analogy kind of went off the rails. But the idea being that in general, you want to be as specific as possible, and your interpretations need to be as specific as possible. The conclusions need to be as specific as possible, so that you don't end up in this situation like Miami Man mentioned, where you start to feel like there's always a study that says one thing and then a study that says another thing. And then another variable on this is some people are just absolute trash at reading studies. And they don't even read the study. They just read the title, and hopefully they read at least the abstract. They pick out a sentence without understanding how the scientist is actually talking about it. And then they just report on it and just throw up the study on the screen and say, well, this study says that. And oftentimes, if you read the study, it's not what the study is saying at all. So that's also a major reason why we have a lot of problems with people not understanding how something can be good one day and bad the next day. It's very context-based, and it's very nuance-based, and it's something that you just have to build over time. And asking further questions, I think, is also important, especially on social media when people are like, well, this bananas cause diabetes. Let's just put that out there. Then you should be asking questions like, well, were the metrics? Were they just consuming bananas? What was the population study? Did they already have diabetes? Like there's so many things that you have to ask from that point on to really get the full context about a story, a study, a story. And people just don't do that. It takes a lot of work to do that kind of stuff. And people just want the answer now. Oh, bananas are bad? Got it. I'll stop consuming bananas. Then the next day, oh, bananas are good? What? Okay, I'll start taking bananas. And they just keep flip-flopping back and forth, back and forth, back and forth. And that's on the presenter, the social media presenter, to be more nuanced, to be a professional, to be better about how they report things. Anyway, I'll leave it at that. I could talk about that for a very long time. So those were the comments. The next topic is ergothioneine and mitochondria. There's this one paper that hadn't been published yet, actually, when I released the content on it. But maybe it has now, maybe it hasn't. I imagine it's quite imminent in terms of its publication. It was a pretty well done paper, especially for it's being submitted to pretty large papers. I'm pretty sure like Nature or Cell or something like that. Science. Anyway, it's about this molecule called ergothioneine and how it could be a boon to mitochondria. So we're constantly focused. It seems like a lot of people are very focused on improving mitochondrial health, and that's fine. That's perfectly understandable. You know, I've studied mitochondria for over a decade when I was doing research work, and this compound ergothioneine was identified through what's known as metabolomics. So essentially it's this massive screen of molecules, and the researchers identify which molecules are most closely attributed to a particular, let's say, metabolic pathway. And if those metabolic pathways converge on, like mitochondria, for example, then you will then say, okay, well, these molecules are the ones that we should be focused on because they're in such high concentration, or they just show up so often in this metabolic pathway. And ergothioneine in this situation was the molecule of interest. So that's what they ended up looking into, and they ended up isolating mitochondria, which was a really cool process. They ended up using tags on mitochondria to then be able to pull the mitochondria out of the cell. So they break the cell open, and then they pull the mitochondria out so that they can isolate the mitochondria and do experiments specifically just on those isolated mitochondria, which is actually an experiment that I've done in the past. Okay, so they've identified this ergothioneine being identified in metabolomics as being highly expressed in mitochondria, especially in muscle, but also applies to other tissues as well. Also, what they discovered is that the mechanism by which ergothioneine provides potential benefit, I guess I'll discuss the benefit real quick. What they also measured when they isolated mitochondria and they measured the activity of these mitochondria, if they had higher ergothioneine or lower ergothioneine, these mitochondria had greater ability to utilize oxygen, which means that there's a better ability for them to produce potentially energy or just to be more active mitochondria. So the activity of mitochondria was increased with ergothioneine present. Okay, so why? Well, the reason why is, and they figured this out too, is that there's this enzyme called MPST. And this MPST enzyme is responsible for adding sulfide molecules, so a particular tag, to proteins that are inside the mitochondria to modulate their activity. And this is called persulfidation. And through this persulfidation, then you can improve the activity or enhance the activity of mitochondria, allowing for better function. Okay, so that's a pretty general understanding of how ergothioneine plays a role in mitochondria, and it plays this role through this MPST enzyme, which has a long name that I refuse to say, mainly because I also don't remember it. And remembering the names of all the enzymes is just, it's futile. There's thousands of them. Okay, then the researchers didn't just discover, okay, ergothioneine is tied to mitochondria, it's through this MPST enzyme. They also wanted to know, well, are there things that we can do that might be able to change the concentration of ergothioneine in our systems, in our body? And what they discovered is, and this is all in animal research to be fair, but the reason why they have to do this kind of stuff in animal research is because, well, it's difficult to do. I mean, you can do a little bit more invasive research with muscle in humans, but it's still far more restrictive than what you can do with animals. So anyway, they exercised the animals, and then they looked at blood ergothioneine levels, and they did confirm that this also happens in humans, that blood ergothioneine levels increase with exercise, and on top of that you get greater localization of MPST, this enzyme, to mitochondria. So you get increases in ergothioneine, and you get increases in the actual localization of the ergothioneine and the MPST to mitochondria. In addition, they also showed that you can supplement with ergothioneine and see the same benefits that you would with exercise. Now, obviously, exercise has tremendous benefits beyond just what ergothioneine does. You know, regardless of what ergothioneine does, exercise has independent effects that are going to affect all kinds of different organ systems. So they're not exactly one-to-one, but if a person, for whatever reason, didn't want to exercise, which is kind of a fundamental aspect of health, but let's just say they didn't want to exercise or be physically active, then ergothioneine supplementation may be an option. Now, this is in preclinical models, so we still need to figure out exactly like the amount and all that good stuff. But here's where the comments came in and were really helpful, because I got a few comments, or one comment that offered some useful takeaways if a person wanted to consume foods that were higher in ergothioneine. Okay, so the first comment, which I thought was hilarious, so I'm adding it here. Jonas had this to say: "What a coincidence! Remember how I didn't want to say the MPST enzyme name? Well, the reason for that is because it's just ridiculous. It's called 3-mecaptopyruvate sulfur transferase." Okay, so Jonas said, "What a coincidence. We considered naming our son to 3-mecaptopyruvate sulfur transferase, but we went with Dexter instead." That's a good one. Yeah, a little easier, but hey, you can always abbreviate MPST. MPST, get over here. Anyway, the useful comment, Jonas, although your comment was useful because it brings joy and laughter to the world. The unseen truth, which is a little ironic considering I'm putting it up on a podcast. I guess you can't see it still, but you can hear it. Porcini mushrooms and yellow oyster mushrooms generally contain the highest amounts of ergothioneine, and also a good amount of glutathione, they mention. So I don't know about the others, but I did look into porcini mushrooms, and that does seem to be the case, that tend to have very high levels, relatively speaking, very high levels of ergothioneine. So if you're a mushroom head, you enjoy eating mushrooms, or just want to add that to a more plant-based style of eating, then there you go. There's an option without needing to supplement, which is really nice. All right, next topic on the agenda is glycine and longevity. All right, so the mechanism by which glycine may extend longevity. I guess I'll quick discuss the result. This was an ITP study, so an interventions testing program study, which means that this was in mice, but it's a little bit more rigorous than other research. And I guess I'll mention real quick that we do mouse studies for longevity research because we simply can't really do, we can do rigorous longevity studies in humans because, well, darn it, we just live too long and we care about freedom. We're not just going to take, one supplement for the rest of our lives or whatever it might be. So the idea here was that the researchers gave glycine to animals from 3 different labs that were genetically different from one another, so heterogeneous mice, which is different from other mouse research in that other mouse research is just given to one group of mice, and they tend to be a lot more genetically similar to one another. So that creates some further problems for trying to translate that research to humans. Of course, mice themselves are not human, so that's another aspect. But even more layers between, translating mouse to human. So the ITP offers a little bit more rigorousness in that you're looking at mice that are genetically diverse, which like humans are. But then on top of that, they're applying it from 3 independent labs. So the 3 independent labs have these huge groups of mice, which really increases the power of the analysis of the number of data points that they're going to be able to get. And then they'll be able to detect an effect if there is one. And then on top of that, because it's 3 independent labs, then you get to see some level of reproducibility or some level of reproducibility between labs, which is sometimes an issue in science. One lab will report something, and then it's like we're getting to the point where we kind of have to wait for at least one other lab to reproduce the results while adding their own flavor to the research, some additions to the research to be certain that the effect is actually real and there isn't something else going on. Anyway, so in these ITP studies, they discovered that glycine supplementation in these mice did extend their lifespan. And the mechanism that was brought up, which, to be fair, I think is a little shaky. I don't think that the mechanism is well understood. But the mechanism was that methionine, which is a different amino acid, so glycine is an amino acid, methionine, a different amino acid, activates the master protein synthesis regulator, mTOR. So mTOR is another one of these enzymes, just like MPST, but mTOR is responsible for cell growth in general. And there's this idea that if mTOR is continuously activated, that you're getting more growth than you might need, and on top of that, that there's this relationship, this association between increased mTOR activity and reduced length of existence. You shorten your lifespan, essentially, if you have elevated mTOR activity for long periods of time. We're talking about years, decades of time. So leucine is the most potent, or one of the most potent activators of mTOR. And there's another one. It starts with an A. I'm not sure if it's aspartate or not, but there's another amino acid that also has this effect on mTOR and activating it. And there's also methionine, which is the one that I'm talking about now. Methionine also activates mTOR. So you've got certain amino acids that are more potent in activating mTOR than others. And where glycine comes in is that glycine can help in the demethylation, if I remember correctly, of methionine, which then allows methionine to be released from the cell. So it goes from a high methionine pool within the cell. Glycine will interact with methionine. I'm assuming this is an enzymatic reaction. Some sort of enzyme that I'm not familiar with plays a role in this, and demethylates methionine and methylates glycine. And glycine does not have this activation effect on mTOR, so therefore you are lowering the pool of methionine, which then reduces the effect, reduces the activity of mTOR, which then would then relate to increased lifespan. Now, this is something I need to look into a lot more to look if mTOR specifically is, I'm sure there's like some genetic knockout animals, or can there be? mTOR is such a potent enzyme, it's kind of hard to just knock it out because it tends to lead, I would guess it probably leads to what's known as embryonic lethality, where before the animal is born, it dies because it simply can't grow. But I could be wrong about that. It's something I need to look into. Maybe that's something that would be really interesting for people. Actually, let me know in the comments, or if you're listening to the podcast, just hop on over to the video version and just leave a comment. Would you be interested in me investigating mTOR's relationship to longevity? Is it really something that we have to constantly knock down, or is it maybe misunderstood? The reason why there's a little bit of doubt in my mind is because, well, one, it's overly simplistic, which the human body is just so complex. It seems a little, there's the Occam's razor aspect of it, like, the simplest explanation is often the correct one. Hopefully that's somewhat correct. But on the other hand, just the human body is so complex, and it's not like we usually just take one thing and then it just fixes all of our problems. So it seems a little dubious in my mind to then point at one enzyme, even if it is a super important enzyme, and say, aha, that protein right there is the reason why we age at the rate that we do. And in addition to that, mTOR activity, stimulating mTOR activity, is also really important for mitigating sarcopenia and for muscle growth and for other positive health outcomes. So it's a little difficult to just pin everything on mTOR. But then again, that also may be why we have this, what I so far still believe, that there's this discrepancy between longevity and health span, that those aren't necessarily always in line with one another. They are up to a point, and then they kind of diverge from one another. But I've covered that in other content. Anyway, if you'd be interested, just let me know. But the idea, the takeaway here, is that glycine seems to improve lifespan. There was another mechanism that was brought up, that glycine can also bind to chloride channels in immune cells, and maybe this applies to other cells as well, but the research has been focused on immune cells. And these immune cells then allow more chloride. I did say chloride earlier, right? I didn't say, okay. Anyway, allow more chloride into the cell. Now, why is that important? The reason why that's important is because chloride is a negative ion. So it reduces the membrane potential of the cell. Let's see, should I go in? Okay, let's go into it. I'll go into a little bit. I think it's a little bit of hand waving by the researchers. Again, like I said, the glycine mechanisms just aren't exactly clear. But to back up one step, your cells are not, they're in this state where they are more negative inside, like the polarity. There's polarity between the outside of the cell and the inside of the cell. The inside of the cell is more negative than the outside of the cell. This is especially true for neurons, especially true for cardiomyocytes, heart cells, but it's also true for other cells as well. Mitochondria, if we want to take one step further, is even more negative than the inside of the cell. And that's what creates the membrane potential for mitochondria, but we're not talking about that. We're talking about keeping positive ions outside of the cell, at least relatively speaking, and keeping negative ions inside the cell, or just keeping a larger proportion of positive ions outside of the cell than inside the cell, because you do have to have some positive ions inside the cell. Now, that said, glycine binds to this transporter that allows or opens chloride into the cell. As I mentioned, chloride is a negative. So it reduces, the idea is that it would reduce the membrane, or it would increase the membrane potential. The membrane potential is the actual difference between the outside and the inside of the cell. But it would reduce the negativity. It would I guess I should say it increases the negativity of the inside of the cell relative to the outside, because you're taking in more of these negative ions into the cell. From there, the idea being that you are less likely to activate the cell, because to activate the cell through a membrane potential change is that you need to increase the negativity within the cell to be more positive. It doesn't have to actually become positive. It just has to hit a threshold. That threshold then allows what's called the depolarization, or the overall complete loss of negativity within the cell. And there's these widespread changes that occur throughout the cell. And this is especially true for neurons and heart cells, heart muscle cells. But exactly how that applies to immune cells, I'm not entirely sure. They just didn't go into it. They just didn't discuss it any further than that. So I don't know exactly how that's all supposed to feed together. It is true that if you increase the membrane potential, it is more difficult for the cell to become activated, but that still doesn't exactly tell me how that would necessarily lead to greater longevity. I mean, in this context, we're talking about immune cells. So maybe if you were to have fewer activated or pro-inflammatory state immune cells, like what we were talking about at the very beginning of the podcast, then maybe that could have an effect at lowering chronic inflammation, which of course would have an effect on longevity. But, it's like I'm as you can tell, I'm already kind of making these like assumptions for the researchers, which just doesn't seem, it's not very grounded in, I mean, it's educated guessing, because I know these things happen, but it doesn't exactly resonate as this is definitely the mechanism. So, but what we can take away is that this study did show that glycine extended lifespan, even if the mechanisms by which it did that are extremely unclear. Okay, the comments. Let me just quick mention one. Dave Fit, are you saturated? Is this person's name. I started taking about 9 grams of glycine a day, divided into 3 servings of 3 grams. At night, I take 3 grams with NAC, that's N-acetylcysteine, before bed. I've noticed a significant improvement in my skin, face is smoother, firmer, etc. I started taking it based on age, loss of glycine, and for joint improvement, etc. But the noticeable skin improvement was a nice bonus. I knew it was good for the skin, just didn't expect to see those results so quickly. Yeah, so nothing about that study actually talked about skin, but as we talked about before, there's a certain level of vanity that comes with, and I mean, even beyond that, I mean, skin health is not just vanity, but it is a major aspect, is vanity, just wanting to look better, which feeds into this whole longevity journey. So anyway, it was interesting to hear the the benefits that Davis has experienced. Again, it's it's not like you're going to get this comparison of like Dave talking about his taking of glycine and another person named, I don't know, Peter, and they started out at the same age, and Dave's been taking glycine for 40 years or 50 years or 80 years. Yeah, let's just say he's 89 years old, and Peter, or whatever I said his name was, died 3 years ago. And then the reason why he died earlier is because of glycine. He didn't take glycine. Like, we just don't have that. There's no way a person can comment and say, my longevity is better because of glycine, because it's impossible to know that without actual scientific studies. And those scientific studies have their kind of arms bound behind their back because of the fact that humans live for such a long time, and we just can't experiment on humans. Those drastic ethics getting in the way of our scientific studies to figure out if glycine is beneficial for longevity. So that's why we use animal studies. Actually, before we move on from glycine, let me also I don't want to go into this nearly as much. I didn't have this planned in the notes, but I did want to quick touch on the fact that I looked at 4 studies on glycine and sleep. And what I discovered was that there was some modest evidence. The studies were pretty well done, but the actual outcomes that they assessed were pretty unsatisfactory for my liking. But the studies that have looked at glycine consumption for sleep have noted, at least across the 4 that I found, a consistent effect of improving sleep with glycine consumption. And there have been a bunch of people that have actually commented. I don't have any of the comments to share because I didn't plan on this, but a lot of people have commented saying, oh yeah, I've been taking glycine for sleep and it's like knocked me out. Other people have said that it's helped them a little bit. I read a comment, I think yesterday or maybe even today, I don't know, days blur together. And that person said that they felt cooler at night after taking glycine, which I found especially interesting. And then there have been a few comments of people saying that it did absolutely nothing for them, which is just the way it is. Science is talking about averages. You're going to have some people that have absolutely no effect and some people that have drastic effects, but the average effect in general was, across the 4 studies, I would say pretty mild, a very mild effect of glycine at improving sleep. So that's like sleep latency, sleep duration, things like that. So pretty important metrics, but don't expect it to just like clear up your insomnia. Oh, although I will say, and I'm well aware this is all anecdote, Ray from my insiders community, which if you haven't joined, please do. In the insiders community mentioned publicly that he had been struggling with sleep for quite some time and had tried a few other things and had no success, but then tried glycine after the video release that where I talked about the studies. And again, I can't stress this enough. The average result was pretty mild, and took it for his insomnia, and apparently it really helped to the point where he felt compelled to like write and comment about it all excitedly. So I was really thrilled to see that. And so anyway, thanks for sharing, Ray. And it might be something to consider. If I remember correctly, the amount that was used in the studies was 3 grams, and they did take it 1 hour before sleep. So results will vary, of course, because that's just the nature of what I just explained earlier. But, if you're the average person and you have a little bit of trouble going to sleep, it might be worth a shot, especially considering, in addition, glycine seems to have other health benefits. So something to consider. Okay, let's get into cocoa and its benefits for cardiovascular disease. So this was based off of a pretty large randomized control trial offering cocoa or placebo to two groups of people, and then followed them, I think, over 2 years, maybe 3 years, maybe 4 years, some number of years between 2 and 6. We'll just say that. I think I'm pretty safe in saying that. And then measured the amounts of cardiovascular events. So events being like myocardial infarction, heart attack, or stroke, or any sort of condition that would be notable, that would land you up in the hospital, for example. Or they also measured cardiovascular deaths. And over that time, they showed that the benefit of just cocoa consumption, keep in mind that the diets were supposed to be the same between the groups, and when you have sample sizes of hundreds and hundreds of people, that whatever differences there are, like let's say if 4 people are strictly vegan or 6 people are strictly ketogenic diet, that gets washed out in the randomization process. One, it gets washed out in the number of participants, but beyond that, it also gets the random chance of randomization. That's the whole point of randomization. Also reduces that issue considerably. So nutrition is to be assumed roughly the same between the groups. That's a little bit less certain when you have smaller groups of individuals, smaller studies. So let's say like 20 people and 20 people. But in this case, we're talking about at least 100, if not way more. I'm pretty sure it was a lot more, actually. And what they found is that cocoa did seem to have a protective effect at reducing cardiovascular deaths. It did not have an effect at cardiovascular events. But I think that they did an adjustment for people who strictly consumed the cocoa every day as they were intended to. So I think they were specifically seeing if they include the entire group that was put on cocoa, told to consume it every day. You have some people that are going to consume it every day. They're very disciplined. And then you're going to have people that are not as disciplined and only take it like 5 days a week, or maybe take it 3 days a week, or just don't take it as much as they're supposed to. And once they establish compliance, which is how many times a week you take it and continuously, then they're able to eliminate people that only had maybe 50% compliance or 65% compliance, because then those individuals are a lot more like the placebo because they're not taking the treatment as they're supposed to. So once they adjusted for those individuals and just only looked at the people that did consume cocoa continuously throughout those years, I believe that they did find an effect for cardiovascular events as well as cardiovascular mortality. But cardiovascular mortality was improved regardless of how often you took it. But there has to be some threshold. So usually it's like at least 80%. Okay, so that was the benefit. And the idea here is that these flavonols that are found inside of cocoa are especially beneficial. So these flavonols, and you can go into the actual videos that show you where I created these like graphics to show you like what enzymes they interact with or not. But the flavonols that come from cocoa reduce the expression or the release of a protein called endothelin 1. So endothelin 1 is released by the endothelial cells. These are cells that line the blood vessels, your blood vessels. So think of like a tube, and the very first lining of that tube is the endothelial cells. These endothelial cells secrete these different molecules, nitric oxide being one, which is another one I'm about to talk about, and endothelin-1 being another one, but they have antithetic effects. So endothelin-1 tends to lead to vasoconstriction, so the smooth muscle cells that are a few layers under the endothelial cells, those are the ones that are actually responsible for constricting the blood vessel. And if you have blood vessel constriction, then that means you have an increase in blood pressure in that area. So if you have higher endothelin-1, typically you have increases in the constriction of the blood vessel because the smooth muscle cells get attached, or endothelin-1 attaches to smooth muscle cells specifically, and then you have the constriction of the smooth muscle cells get activated to then constrict. So flavanols from cocoa reduce the amount of endothelin-1 that's released, which would then reduce the amount of constriction that's occurring at the arteries. The second thing that it does as well is it stimulates nitric oxide. And there are a bunch of mechanisms by which it does that. There's some like specific flavanols that are in cocoa that have some unique effects at increasing nitric oxide. So they might they inhibit arginase, for example, which is an enzyme that degrades nitric oxide. Nitric oxide is kind of the opposite of endothelin-1 in that if you are increasing your nitric oxide release from the endothelial cells, those can also bind to the smooth muscle cells and have them relax. So they become activated in the opposite way, as in they relax, and therefore the lumen, or the opening of the blood vessel, opens up. So arginase, I believe I mentioned, is the enzyme that degrades it. So if these flavanols can inhibit this arginase, then that means you have more on the back end of nitric oxide. It also tends to increase, I believe it's an increase in a molecule inside of the cells called cyclic GMP. The cyclic GMP is a signaling molecule, a very important signaling molecule. And cyclic GMP will allow more nitric oxide to be produced as well. I'm hoping I'm remembering the right direction of that, if that's an increase in cyclic GMP. Anyway, so those are a few of the mechanisms by which, and then the overall effect that cocoa had on cardiovascular mortality. But one more warning, or one more thing I should say, is a warning, is if you did end up deciding that you wanted to consume cocoa for your overall health, because I would imagine that cocoa probably has benefits beyond just your heart health, you would want to make sure that you get it tested. And it's not like you yourself have to get it tested. There are different websites and companies and whatnot that will do kind of independent testing, and there are certain regulatory bodies now that they're independent regulatory bodies that will test a lot of these different molecules, these different supplements that are out there. And cocoa is one that I probably would not start consuming cocoa, and this has to be at least 70% cocoa if you're going to eat it in like chocolate bars or whatever it might be. But if you use it as a powder, it should be 70% cocoa or higher. I mean, 100% is certainly better. It should also be undutched, which I'm about to get into as well. And when you have it tested, you want to look for metal poisoning. So like arsenic, cadmium, I believe, was another one of the poisons, and lead is another one of the poisons. So you want to make sure that it has the lowest levels of those, because especially I believe cadmium is the one that really builds up in your system. So you want to make sure that you're not over consuming on cocoa for the health benefits, and then you end up shooting yourself in the foot or maybe even in the face because you're consuming these heavy metals. Anyway, something to consider. So definitely get it checked if you decide to try the cocoa bandwagon. Some comments on this. Skeptical Caveman. For those who drink coffee, just mix a tablespoon of raw cocoa powder into your coffee. Yeah, I mean, that's the way that a lot of people take, like collagen, for example. You can do it that way with collagen, cocoa powder. I do wonder if the heat from the coffee, I suppose maybe it's not going to be that extremely hot. If that might change things, I don't know, but I'm skeptical. Skeptical Caveman. Another comment, Nagana said, the undutched chocolate that you reference is non-alkalized chocolate. Alkalized chocolate is less bitter and is used in milk chocolate. Yeah, we're definitely not talking about milk chocolate. If you are looking to buy the beneficial chocolate, vendors such as Amazon do not use the term undutched, so you should look for non-alkalized cocoa powder. Studies I've seen regarding the risk of heavy metal, namely lead, in cocoa have shown that this is not naturally occurring. Rather, cocoa beans dried next to roads accumulate lead from vehicle exhaust. See, these kinds of things are things that I don't know. But it is true that when I've looked, Amazon did not have a great selection of these kinds of undutched or non-alkalized chocolates. So in terms of like where the lead comes from, that I have absolutely no idea. I tend to just worry about, is it there? How do I vet for it? Is it going to affect my health? And that's it. Everything beyond that is completely out of my scope. So I just I just let it go. But that seems reasonable. But if it's true, I don't know. All right, let's move on to the next topic, which is glucose spikes causing cardiovascular disease. The reason why I picked this to cover was because there was this Jessie, the glucose goddess, was blowing up on social media. People were asking about this craze about glucose spikes. Should we be worried about them? And this is a prime example of what I'm talking about when I'm talking about being more specific about questions and outcomes and stuff, is because saying, hey, is this a problem? That is such a general thing. Like you could focus many years on just one of these areas. Glucose spikes. Are they a problem? Are you talking about cancer? Are you talking about particular types of cancer? Are you talking about heart disease? Are you talking about dementia? It's all these different things, and all, like, to do a proper investigation of this stuff, you have to do a lot of work and open up many, many studies. So anyway, I chose cardiovascular disease, and that's probably an area that has the most research, and even that really needs a lot more research. And I'll explain why, and I'll offer a little bit of context to the glucose spike craze. But really, we do need more research on this. So the mechanism that's been proposed in some of the research that I looked over is that if glucose spikes upwards, that you have the production of these AGEs, or essentially these end byproducts of glucose being attached. So these advanced glycating end products. Wow, that was having trouble coming to me. These AGEs are these glucose molecules, just like you would, consume them, and they get non-enzymatically bound or attached, chemically bound to any number of different things inside of your body. If that's proteins, if that's fats, whatever it is, it can bind to them if it's in a high enough concentration. That's the idea of this glucose spike. And then these AGEs are related to any number of different disease states, diabetes, cardiovascular disease, and so on. So how do they actually have an effect? I mean, once you produce them, I feel like a lot of people it seems to me that people say, well, we have to get rid of these glycating end products. So that's fine. But what's the mechanism? Like, why? Now we have them. It's like if we have a large amount of chylomicrons, which are these the things that we package a bunch of fats into. When we consume a large amount of fat, that fat gets repackaged in our intestines and gets sent out in these chylomicrons. So just because you have a large amount of chylomicrons, what would be the mechanism for their damaging of your body? And in this circumstance, we're talking about AGEs. It's just seemed to me on social media that people just kind of say, well, we just got to get rid of AGEs. And maybe somebody will say, oh, they increase oxidative stress, which is some stuff that we've talked about earlier. But that's kind of unsatisfactory for me. So I did look into some of the mechanisms, and one of the mechanisms is through this RAGE pathway, which I think is just one of the coolest names for a pathway. The RAGE pathway is activated. So the RAGE receptor is bound on the exterior of the receptor. So the receptor is embedded into the cell membrane. You have a cell membrane, you have a receptor that's inside of it, but it goes through, so it's a transmembrane receptor, meaning that there's a section of the receptor that's in the actual fatty layer of the cell membrane, and then it has an inside portion that's primarily proteins, and then it's got an external section. So the intracellular section of the receptor and the extracellular section of the receptor. And the AGE will bind to the extracellular section of the receptor. And when it binds to this RAGE receptor, it changes the conformation or the shape of the internal section of the receptor, this intracellular section, which then leads to the activation of an enzyme that I very briefly teased at the very beginning of this podcast called NADPH. This NADPH enzyme, oxidase enzyme, ends up generating oxidative molecules. So it generates an overabundant level of these potentially damaging molecules. So that's one of the ways that AGEs are proposed to have a negative effect on the cell. And of course, this multiplies out millions and millions of times to apply to your entire system. Another way that it does that is if it binds to the RAGE pathway, and the same thing happens, but it activates what's called the ERK pathway, the ERK pathway. And what that is a grouping of proteins, and they go through this cascade. So one protein gets activated, then another protein gets activated, then another protein gets activated, and so on and so forth, until eventually you get to this kind of terminal protein that goes into the nucleus of your cells, binds to different genes, and then leads to the expression of those genes. Now, those genes, in this instance, are pro-inflammatory gene expression. So you're increasing, again, going back to like the cytokines, for example, you're increasing the cytokines, or you might increase the amount of ICAM activity. ICAM is a protein that your immune cells can bind to and then stay in that region, and they can also invade past the cells if they're able to stop and anchor to these ICAM proteins, and then they go below your endothelium, and then you get this this kind of propulsion of inflammation occurring within your arteries. So this pathway, the RAGE receptor, gets activated, and then downstream it activates a protein called RAS, which activates a protein called MEK, which activates a protein called ERK. And that ERK protein is the one that ends up influencing the gene expression. So those are a few of the pathways, some of the mechanisms, but there are certainly others that are out there as well. I don't want you to think that, I cover everything in these pathways, but it gives you some idea of, okay, there's complexity here, and there's many ways that these molecules can affect our cells. Okay, so that's the idea. Now, that's the mechanism, and I don't think anybody would argue that AGEs are good for you. I think that they're pretty well established to be a negative health consequence, but do glucose spikes themselves, that let's say they do generate AGEs, do they actually generate them in such an outsized way that ultimately leads to increases in cardiovascular disease? And there have been some experimental human studies that indicate that there's an increase in oxidation, so with the reactive oxygen species we've talked about, as well as reduced flow-mediated dilation, so the amount of blood flow that can flow through a artery. So remember when I talked about with nitric oxide and endothelin-1, the enlarging of the artery allows more blood flow, obviously, through the lumen, the area that actually allows the blood flow through. And if you constrict it, then you have less. And with flow-mediated dilation, you want to have the most amount of dilation, or the most amount of opening of that artery, which then allows you to feel like, okay, you've got a lot of flexibility there. That's a great sign for overall cardiovascular health. Now, it's not the best metric to just use that kind of evidence, because there are other areas, like if you consume, I believe there are certain fats that people can consume that lead to reduced flow-mediated dilation. There are also effects of exercise. So acute exercise can decrease flow-mediated dilation, which both of those would then seem like it's a bad thing. And the reality is that you have to look at long-term data, not just look at flow-mediated dilation, because flow-mediated dilation in an acute sense can sometimes look worse. And then a day later, a week later, months later, flow-mediated dilation on an average sense improves. So those are the kinds of things that you really have to consider. So if we step away from those really short-term experiments that are done over maybe a day, but they are in humans, which is nice, then we start looking at some pharmaceutical methods for reducing glucose spikes. And one of those is repaglinide. So there's this comparison study of repaglinide versus glyburide. And both of those, their aim is to reduce blood sugar, but they do it through two different mechanisms. I don't remember the exact mechanism of repaglinide versus glyburide, but the general gist was that repaglinide was the one that was supposed to stomp out or reduce the actual glucose spikes themselves. And glyburide was the one that lowers your overall blood sugar over time, but does not necessarily stomp out the glucose spike. And they do show some data that the glucose spike is smaller in the repaglinide condition. And in that study, when using repaglinide, glucose spikes are lower, and they also looked at intima media thickness, so looking at the thickness of the wall of your blood vessels. And they show that there's a reduction in the progression of this intima media thickness. So the thicker it is, the worse
your health, because that means that there's atherosclerosis occurring of some sort. So the fact that it's reduced is a huge benefit in favor of repaglinide, which would then mean, okay, well then we're also addressing this glucose spike. And yet, what was interesting is that the fasting glucose was lower in the glyburide condition, because what we're trying to do at this point is try to separate out, if you have just elevated blood glucose levels, that is a cardiovascular disease risk. So how do we separate out the specific risk of the glucose spike versus the overall increased blood sugar that you have on a consistent basis? If you'd never had a spike, but you had high blood sugar, or if you have glucose spikes, but you don't have high blood sugar, or if you have both, how do you distinguish between those? And that's really difficult to do because they're really intimately tied with one another. And if you were to switch to like nutrition to try to achieve that, now you're introducing so many other variables that could be having an effect on the glucose spikes. And then, of course, you're not going to be measuring like intima thickness. If you're getting to that level of granularity, it becomes a lot more difficult to do studies. So anyway, this study showed that with the glyburide condition, which did not reduce glucose spikes, that one had lower fasting glucose and yet still had worse intima media thickness compared to the repaglinide condition, which did dampen down the glucose spikes, which would indicate then that it might not be because of the fasting glucose levels, but because of the glucose spikes, because the repaglinide condition had higher fasting glucose and they were still able to see improvements in their intima media thickness. That said, however, the wrench in here is that inflammation was also lower in the repaglinide condition. So the C-reactive protein measurements were improved in the repaglinide condition. So now you're seeing, okay, now is it the glucose spikes, or is it the fact that they were able to reduce the inflammation? Or these could also be very well tied together. And maybe, but now it's the chicken and the egg. Which one comes first? Is it the reduced inflammation that came first, or is it the glucose spike? So there's just so many variables that are moving all at once. And of course, this study also didn't have a placebo condition, didn't have a control condition. So that makes the interpretation even harder. On the other hand, there was another study looking at acarbose, which is essentially delays the uptake of glucose or carbohydrates into the bloodstream. And that one did reduce heart attacks compared to placebo. But the problem with that, again, is that, so they did have a placebo group, that's great. But on the other hand, the problem is that you're still affecting many other things. Usually you're not just affecting the glucose spikes, you're affecting multiple other factors. So which one of those factors is actually the one that's causing this improvement? So overall, and then there are some other studies that I went over as well, but none of them give clear, definitive evidence that glucose spikes absolutely are this horrendous thing. So overall, we need more studies. This is one that is just really up in the air, really wishy-washy. But something else I wanted to add here is that healthy individuals, I did find a study that showed that healthy individuals have glucose spikes above 140 milligrams per deciliter for less than 1% of 24 hours. So now for glucose spikes to be that detrimental to your health, this thing would have to be incredibly potent in that 1% to have an effect for the other 99% of your day. I was about to say 23% of your day, getting 23 hours and 99% confused. For the other 99% of your day, which if that isn't experiencing really high glucose spikes, which by the way, 140 milligrams per deciliter is not like supremely high by any means, but it is higher than your fasting levels. So you'd have to incur so much damage from that 1% moment for you to psychologically have to worry about this to a huge degree. And I just don't think that healthy individuals that don't have diabetes or not pre-diabetic really have to deal with worrying about this stuff to any great degree. At the current moment, I'll just put it this way, at the current moment, the evidence is so shaky. I would focus on so many other factors like physical activity, just getting your nutrition in check, making sure your body weight is normal, like those kinds of things, accruing muscle. Those things are far more important than sitting there making sure that your glucose spike doesn't just barely reach into 145 milligrams per deciliter one time in your day or two times in your day. It's just like that is so much work to worry about something that takes up such little amounts of your overall day, which also means that it likely makes minimal impact. That's my guess, that it probably makes minimal impact for healthy individuals. It might be a different story for people that already have chronically elevated blood sugar levels. But let's say you did want to reduce it, the glucose spikes, you should be focused on the actual spikes of the magnitude of the effect and the duration of the effect. So if you have a huge glucose spike, but it sharply goes back down to the kind of normal levels, then that might be better, that probably is better, than having a sudden rise and then having that last for 60 minutes, 90 minutes, or whatever it might be. Oh, one more thing that I forgot to mention that I don't have in my notes. Keep in mind that when we're measuring glucose spikes, we're actually not measuring immediately after consuming the meal. We're not measuring it at the highest point that it hits. A lot of the measurements, at least by, if I remember correctly, by the oral glucose tolerance test is at least 2 hours. But I think most measurements are at least like 90 minutes or something like that. So a lot of the benchmarks that are used, I'll put it this way, the benchmarks that are used in studies are set at, I believe, 90 minutes after consumption of food. And what people are getting really freaked out about is 30 minutes or 45 minutes. It's like, of course, that's going to be higher. And yet the studies are focused on 90 minutes. So you can't make that equivalent because they're two completely different times. And for glucose spikes, that's a major, major factor to get the timing correctly for when you're measuring. So that's just another thing to consider. So some comments. Billy Boy 66 said, I stopped eating sugar, processed foods, seed oils, and all I can say is that it's life-changing. Okay. Down 50 pounds in 11 months and exercising like a mofo. Okay. Well, congrats. I mean, seriously, that is highly congratulatory. 50 pounds in any amount of time. 11 months is great, but any amount of time is really fantastic. I'm 58 years old. I couldn't find the motivation to exercise until I stopped eating American junk. Unfortunately, I have ASCVD, which is cardiovascular disease. Has a CAC score of 600, discovered a few months ago, so pretty elevated. And so now I'm simply trying to stop the progression of plaque. I eat foods with one ingredient, that's it, mostly meat and veg. Wish me luck, Nick. Well, Billy Boy, I wish you luck, of course, as always. But also, really commendable. Losing 50 pounds, that'll make a big difference on your health. Huge difference, which I don't need to tell you. You already said it, life-changing. That's how I would describe it too. And being able to stop the progression of the plaque, yeah, I think that's very feasible. The one takeaway I would say is that usually when people comment in and say, oh, I did this change, and it led to this outcome. Usually that's not in a systematic way. So like this doesn't necessarily tell me anything about the glucose spikes. So stopping eating sugar, right, that would contribute significantly to glucose spikes. Processed foods maybe as well. Seed oils, probably not, even though people feel very strongly about seed oils. I mean, they are fats, so seeing a glucose spike from a fat would be a little weird, or at least a significant one. So, and the fact that you're changing so many different things plus exercising, it probably isn't the glucose spikes that are actually causing all these benefits. But ultimately, we don't really care, because what you're doing is you're prioritizing what's the most important, which is doing the fundamentals, cutting out processed foods, making sure that you're exercising, eating less, which is just going to be a function from eating less processed foods. Sticking to foods with one ingredient. I mean, maybe you don't have to get that that aggressive with it, but if it works for you and if you can stick with it, that's great. 5 ingredients, 3 ingredients. There's all kinds of different benchmarks that people like to create. Either way, whatever works. The lowest number of ingredients is probably going to be great, simply because you're sticking to a food that is not going through a ton of processing, that's not going to be having a ton of things added to it. Like the processed foods that have the ingredients that are like 50 different ingredients for all kinds of different reasons, which is something I need to create a lot more content on, looking at processed foods and the effect that it has on our health. Some of it is pretty obvious, but I'd be really interested in some of the mechanisms as well that go beyond I eat too much. I mean, that's pretty obvious. Okay, Dennis Forbes, excellent summary. Thank you. It's also worth noting, and they were talking about one of my videos, not my podcast, which clearly is not summarized. It's also worth noting that one of the reasons that there is an absence of compelling data is for simple functional reasons. The appearance of continuous glucose monitors is a very recent invention, and any sort of large-scale, long-term study cannot possibly come out for probably years. So in a few years, we may learn that controlling glucose spikes is the key to health, or we won't. Yeah, that's a fantastic point. That's true. Glucose monitors, at least the way that we've been using them recently with so many people trying them, has not been around for a very long time. And when we are assessing things like ASCVD, cardiovascular disease, which takes years and if not decades to really be prominent, especially when you're trying to measure events, actual cardiovascular events, it's difficult to assess that, or it's basically impossible to assess that if you just don't have the right time horizon with this new technology. So in this case, glucose monitors. So it's a great point. In the future, we might be able to start teasing out. There are going to be correlations, to be clear. I don't think we're going to get randomized control trials on this kind of stuff. This is going to have to get teased out just through correlation. And ultimately then if we did say that glucose spikes were dangerous, then I would then wonder, well, how much of an effect is it going to have? So some things to consider. All right, the next topic to go over, multivitamins for cognition. I just had I don't have any comments for this. I don't really have a mechanism that I needed to cover for this one, but I did want to quick mention that I covered a study, I think it's called the Cosmos studies, Cosmos Mind, as well as Cosmos Clinic studies. And they looked at multivitamin use in those over the age of 60. And over 2 years in healthy, non-demented individuals, which I actually like the fact that they focused on non-demented individuals, so people that are mentally pretty healthy, mainly because I'd like to then see if they can pick up an effect of multivitamin use. And what they found is that, yes, they were able to detect an effect. However, I will point out, and this was actually something I did not emphasize in the video, which is a shortcoming of the video. I did end up adding an amendment or a reminder, just a note to people. And because some other people had mentioned this, which I thought was an excellent point, that we may be seeing a very small effect. So the study showed a very, very small effect in benefit of taking multivitamins for your cognitive health over those 2 years. What I failed to mention is that it's possible that over a longer time horizon that you would see that effect increase even more. And on top of that, I should point out that when you're talking about healthy individuals, it's difficult to then say, okay, you're going to see a huge effect of anything, really, literally anything. How are you going to see a huge effect when people are already do not have any sort of mental pathology or brain pathology? It's a completely different story if a person has deficiencies and then you supplement those deficiencies and then they get remarkably better over, let's say, a year or two. That's a different story, and you actually don't need as long of a time horizon to detect an effect. So even though the effect is very mild in this circumstance, it doesn't necessarily mean that it's useless. And on top of that, multivitamins have other usefulness about them. I know that some people I think some people have sent me some stuff on multivitamins increasing risk of mortality. I think I briefly perused those studies. That's not exactly what the studies were saying. Maybe I'll cover that in some future work once I've actually looked into it in far more depth. But again, we have to be focused in on the outcome of interest. So multivitamins on cognition. And in this case, it is a net benefit, but it's not going to be a huge effect. But since you're maybe already taking multivitamins for other things, just to make sure that your health is okay, it's really not an added cost if you're already taking it, and it's just a little bit of an added reason to take multivitamins. And we'll see if that pans out in some of the other research. I mean, maybe multivitamins are useless in other metrics. I believe I looked at one video by Johnny Harris looking at multivitamin use on the effects that it had on other outcomes of health. And I didn't see anything overly striking there, that it basically had a relatively neutral effect, unless you hyperdosed on vitamin A, I want to say. But then we're not talking about multivitamins. Now we're talking about specific vitamin regimens, which is not what we're talking about here. Anyway, point is, very small benefit on cognition, but the benefit was there. And that's for people over the age of 60. Okay, on to the next one. Let's go on to red light therapy on skin aging and mitochondria. Oh man, I gotta be honest, I did not care about this topic really at all. I did this simply because I got so many requests to do it, and I try to listen to what people want. Sometimes I do things because I'm interested in them, and other times I do them because there seems to be a groundswell of people that are just really interested in a topic. So red light therapy is one of them, and it's a confusing one. It's really a confusing one. So I looked at two different metrics. I looked at skin aging and I looked at mitochondria. Got a bunch of complaints from people saying, well, why don't you focus on inflammation? Why don't you focus on cancer? Why don't you focus on eye health? Why don't you focus, like all these different things. But again, I just can't stress enough that it really, you can't have a shot, like a buckshot approach to this stuff. You can't just say, well, there's this one study in eye health, and then I'm going to go over to this other study that looks at cancer, It just doesn't work. The best way to get a robust mindset on evidence is to aggregate a bunch of studies on one topic, cover that, then aggregate another grouping of studies on another topic, cover that, and so on and so forth. And I mean, this kind of stuff, these investigations just take me weeks to do. And although there seemed to be a lot of people that were really interested in red light therapy, the videos actually didn't do that well. Even one that was covering mitochondria, which is an area that people really find very interesting. So considering it's a topic I don't really care that much about, considering it doesn't do that well, and each video, full disclosure, cost me hundreds of dollars to make. And I just don't really see the appeal of then looking at inflammation, cancer, all these different things, especially when the science is not that great. And that's kind of what I'm getting into here with the skin aging. Again, the reason why I picked skin aging was because I was mildly interested in it. And on the other hand, I was also trying to focus a little bit more on the aging aspect, which seems to be of great interest to people. So, yeah, the studies on skin aging were pretty shoddy. I mean, almost none of them had, I think none of them actually had placebo groups, which I realize is difficult to do, but there might be ways that you can figure out a placebo condition. Not to mention, you can also use questionnaires and figure out how effective the placebo was, which is something else that I think that we often fail to address in research, that you can just ask and see, hey, which condition were you on? Do you think that you were on? And the participant can then answer. And if you have like 80% of people that get the right answer of like, yes, I was on placebo, then okay, maybe the placebo wasn't very effective. People know that they were on placebo. Or, if they, yeah, I mean, you get the idea. So anyway, it is definitely more difficult to do more placebo trials, but it doesn't even necessarily have to be placebo. It can also be like another type of treatment, and then have like a regular control group. And then maybe between those two, you'd be able to tease out differences against the actual red light therapy group. So anyway, I think there was like maybe one or two studies that used like an actual control group, and that control group just didn't experience anything. So they were just measured and nothing was applied to them. And then they were measured again at the end of the study, which is just like, it's just a comparator. That's all it is. It's not really a true control group, if you want to put it that way. So and then there were some studies in cells. There were some studies in animals that's the mitochondria side. But for the skin side, most of the studies were also industry funded. One study was industry funded. One of the researchers was the CEO of a red light therapy mask. And on top of that, they did a bunch of experiments and did the wrong statistics on all of their experiments. So I had half a mind to just kind of chuck that entire study. Not again because of the potential conflicts of interest, but because of the statistics were wrongly applied to on all their experiments, which then kind of gives me this sense of like, okay, maybe if you mess up the statistics on one, that makes some sense. But if you mess it up on all of them, it just kind of gives this sense of like maybe carelessness. I don't know. Anyway, the point being that overall the skin studies, if you want to believe that red light therapy works, all of them showed that red light therapy was beneficial to your skin. So great. But if you only follow scientific rigor and you want to always stick to control groups and placebo groups and all that stuff, you would be wholly unconvinced by the data on red light therapy for skin. Now, the next one is to go on mitochondria. And here, what's intriguing is that you can do a little bit more with some more experimental research like in vitro, so like cell work or animal work. And with animals, I mean, as far as I'm aware, I'm not a veterinarian, or I don't really understand animal psyche that well. But if you were to apply like a red light therapy to animals and then apply like a different type of color therapy, of light therapy that doesn't penetrate the cells, then the animals definitely won't know. And you could probably do this with humans as well, because there's different wavelengths of light, as I understand it. And red light therapy, I believe, is 600 to 700. I also looked at near infrared, near red light therapy as well. And all of those studies seem to indicate there was a benefit to mitochondria. But that benefit is conditional on the length and intensity of the light. And if you're expecting me to get into specifics on exactly what length and exactly what intensity, that stuff is really up in the air, because the studies use a wide range of intensities, a wide range of different durations, and different styles of how they did red light therapy. So I'd just be taking just a complete shot in the dark if I said, oh, this is the exact temperature. But what I can say is that doing it at the highest intensity possible actually led to worse mitochondrial outcomes. So there's a relatively wide band there, where if you use like moderate intensity red light therapy, however you define moderate, I can't define that for you, unfortunately, based on the current evidence. But moderate red light therapy led to improvements in ATP generation by mitochondria, as well as other metrics of mitochondrial activity. Yet going more intense eliminated that benefit. And if you were too low, if you used too low of intensity, that actually worsened mitochondrial health or mitochondrial function. So, I mean, like I said, all over the place. Now, in terms of the mechanisms, it seems to affect mitochondria by energizing complex 4. So you have these different complexes that are found inside. Mitochondria have two membranes. On the inner membrane, the second membrane, there's these different proteins, and these proteins are the ones that are responsible for ATP generation. And they have to work together. So all 5 proteins work together, as in they exchange electrons, to eventually lead complex 5, or the ATP synthase, to generate ATP. So without those 5 complexes working efficiently, then you're not generating as much ATP. So the idea being that red light therapy would directly affect complex 4, because complex 4 binds to oxygen, and oxygen is one of the critical components for this whole like electron system. I'm being a little general here, mainly because I just don't want to get into the specifics of it, because it just doesn't add that much to our understanding. If you want more specifics, you can check out the video, but there's something unique about complex 4, and that unique aspect is the fact that it interacts with oxygen, does something to oxygen, which then allows this whole system to work. That's what you need to get across from this. And red light therapy increases the ability for this complex 4 to function. Additionally, it seems to also affect complex 5, and I should say on complex 4, it also, if I understood this correctly, which I think I did, but the exact mechanism, like the sub-molecular mechanism, which is something that I think I would probably need to consult with like a physicist and maybe a structural biologist to be able to figure exactly how this happens. But red light therapy can also encourage the dissociation of molecules that would otherwise block oxygen's interaction with complex 4. So that was that was a more direct mechanism of what I was just talking about. It also increases the speed at which ATP synthase, complex 5, is able to generate ATP. So, and then there's something called retrograde signaling, that if you increase the efficiency of mitochondria and they end up generating more ATP, that ends up kind of bleeding into the cytosol, so outside of the mitochondria. And then the changes in the amounts, the concentrations of these molecules inside the cytosol now allows all these different enzymes and these different genes and all these different systems within the cell to change how they react or how they behave. And that's so instead of them affecting mitochondria are affecting all these different systems. That's why it's called retrograde signaling. So there's a bunch of other proposed mechanisms that are just like, they just take so much effort to understand, and they also require, I think, degrees that I do not possess. I did consult with my dad, who has his PhD in physics, on some of this, and we talked about it for a little while. The mechanisms that I just went over seem to be pretty solid, although the science behind them is just, it's the type of science that is starting to get to the fringes of my level of expertise. So it's probably an area that I can't assess as well as somebody who has a degree in chemistry maybe, or physics, maybe biophysics, and structural biology. Anywhere in that realm, which is a little bit to the left of where I am, would be able to give a little bit more detail on that. Okay, so some comments. I guess the big takeaway here, just to close this out, red light therapy seems to improve mitochondrial function. And I'm a little bit more convinced of that than I am of the anti-aging skin data. I'm still a little shaky on it, but it's better than I initially expected. I'll just put it that way. Okay, comments. Zappy O, which, by the way, Zappy always has some fantastic comments. Very funny. I've laughed multiple times. Zappy O says, should I go to the red light district to obtain these benefits? No, Zappy. You do not need to go to the red light district. For anybody that doesn't understand that joke, I'm not going to help you. Sorry. Niels 5008. I like this video. Thank you. Not talking about the podcast, obviously. I swear by my red light therapy panel. It really improved my hair growth by a lot. It really is night and day from when I started using it a few months ago. All my other treatments only affected the hair edge, but red light also affects regions much farther away from my hairline. It really is something else. Okay, cool. Well, thanks for sharing. And another anecdotal piece is from Ceslasosceles. I am not saying that again. I have never felt more repeatable, robust, consistent, and efficacious effect for my well-being that I have from consistent red light therapy since I started using it 1 year ago. And at first it felt almost negative, like detox, but soon turned into one of the most enjoyable lifestyle staples I've incorporated thus far. The dose makes the poison or the medicine. Red light has been no different for me. Well, again, it's great. It's encouraging to hear these things. Obviously, these aren't like mitochondrial assessments or skin assessments or anything like that. But hey, I mean, I guess if you're doing something and you feel like you're getting that much benefit from it. I've said this before, but who cares about the mechanism of like, oh, is it actually working? I mean, it's working for you, so great. That's really wonderful. Okay, well, I teased it much earlier, actually, in our very first topic, looking at astaxanthin. Now we're going to talk about the sister molecules, zeaxanthin and lutein for eye health. So the mechanisms are largely the same, so they are potent antioxidants because they have a similar structure to astaxanthin, a little bit chemically different, but for the most part, largely the same, which allows them to quench these damaging molecules, the reactive oxygen species that we talked about. But on top of that, they actually get embedded into the cell membrane. So we've been talking about the cell membrane a good amount. It's what separates the exterior of the cell from the internal components of the cell. And zeaxanthin and lutein can actually embed themselves or be found inside the membrane of the cell itself. And what that allows is, first of all, protection from any sort of external factors that might increase, like reactive oxygen species. But what it also does is it attracts antioxidant binding proteins from the cytosol, so from other areas of the cytosol, so that's the kind of the open area within the cell. These proteins will be attracted to zeaxanthin and lutein and find themselves at the cell membrane, which then localizes these antioxidant proteins to that region right on the surface of the cell, which not on the actual outside of the cell, but just below the inside of the cell. And that can also act as a layer of protection for the other sections of the cell. So I thought that was pretty cool, and probably there are other mechanisms as well. What's interesting too is that lutein is found all over the retina, so it's found all over that back part of the eye. On the other hand, zeaxanthin accumulates directly in the macula, which is a particular specific section of the eye. So zeaxanthin is highly concentrated in that area, which is what also makes zeaxanthin an especially unique molecule to the eye. If I remember correctly, those two molecules, especially zeaxanthin, end up almost not completely, but a large part of it gets just taken up in the eye. So they end up finding themselves and attracted to, in a manner of speaking, to the eyes. So that's what mechanistically makes them such interesting molecules that are unique from other molecules that might have similar effects, antioxidant effects, like astaxanthin. I don't actually know yet if astaxanthin goes to the eye, but even if it does it have the same level of specificity that zeaxanthin and some other derivatives like mesozeaxanthin and lutein do? And if you were to pair astaxanthin with those, would you actually see better results? My impression is probably not. It seems to me, and this is just educated guesswork, to be clear, it seems to me that maybe astaxanthin would be better for the totality of the body, and for these two other molecules, lutein is also heavily implicated in the brain, or in other parts of the brain, I should say. But zeaxanthin would be then more particular to the eye health. So, do they actually help in eye health? Well, I went over a number of studies, 6 or 7 studies, something like that, and the evidence is quite good, actually, in the pairing of zeaxanthin and lutein to improve vision and reduce the risk of macular degeneration. Now, something that I made sure of, one of the tests that's commonly used for eye health is to use an MPOD test, which is macula pigment optical density test, something in that realm. And so that was used in a lot of studies. But the problem that I have with that test is that you're just measuring the overall density or the overall health of, let's say, the macula, but that doesn't actually give you functional measurements. It doesn't say, oh, now I can actually see better, like some of the comments that we talked about earlier, like with astaxanthin. And the only way to do that is to actually do functional eye tests over time. So I believe I did find one or two studies that did look at functional eye tests and did indicate that there were some mild improvements in eye function from the continued consumption of both zeaxanthin and lutein. I also don't think I found any studies that separated the two. They were always consumed in combination. And that might be because of their localization. Lutein is found across the entire retina, while zeaxanthin is, not that it's not found anywhere else, but it's largely heavily focused on that one region. So that was so zeaxanthin and lutein for eye health. Looks like a good thumbs up on that one. Joseph Wu says, I've been taking them, and recently I've been seeing my neighbors through the wall, so I had to stop. One thing that I really love about Physionic, beyond being able to talk about physiology, beyond being able to do what I love, beyond being able to read research and just educate myself to a massive degree and to help other people with their health, is I have been a lot more myself, just with jokes and whatnot, which I know make people cringe. And they crack me up even when they make people cringe. But it also attracts people like this. Joseph Wu. I mean, that was a good one. That was a really good one. I've been taking them, and recently I've been seeing my neighbors through the wall, so I had to stop. Okay, Superman. All right, Seosin, a more serious one. I've taken lutein, zeaxanthin, astaxanthin, taurine for 2 years, and I've stopped needing glasses from a low prescription. Yeah, so, I mean, we do know, like I said, there were a few studies that showed improvements in eye function. So could you see some reversal of like low prescriptions? Yeah, possibly. I don't think that, I'm going to stop needing my glasses, but still it's, that alone is pretty impressive, just to take some supplements, and you don't have to get them from supplements. I believe the main source, if I remember correctly, of these molecules was kale. And then there are a few others that have much lower levels, but some at least appreciable levels of these molecules. Is lutein found in eggs? I think so. That's also another source, but I don't know the exact amounts of each one. I do know kale was really high up. It was probably number one on the list. So you don't have to supplement with them, but if you wanted to, you easily could. Anyway, impressive results. Now, on to the next topic, omega-3s on cardiovascular disease. I have quite a lot to say on this because I did a ton of research on this. Okay, the omega-3s, omega-3 fats, are polyunsaturated fats that have been linked to a number of different health outcomes, positive health outcomes. But I was especially interested in the effect that it had on cardiovascular disease. And I think in the past I've discussed their effect on the brain. So DHA and EPA, so docosahexaenoic acid and icosapentaenoic acid, those are the two main omega-3s that people talk about, have different effects in changing your cell structure. So they have a number of mechanisms by which they've been proposed to help in cardiovascular disease. And we'll go into the actual clinical results, which is a little bit less certain than one might initially imagine. and then it becomes a little bit more certain once you understand some of the nuances. I'll explain what that all means in just a bit. So let's return back to your cell membrane. In your cell membrane, you have many different molecules that are embedded. You have these phospholipids, you have different proteins, you have glucose, like different sugar molecules, like saccharides that are stitched together. You have all these different molecules that are found in your cell membrane. Another one is called cholesterol, these like cholesterol domains. And these cholesterol domains, when they get embedded into the cell membrane, they tend to stiffen the cell membrane. That's not necessarily a bad thing. I don't want you to think stiffening is always bad. It's just you usually want to have some level of balance between, some level of flexibility and some level of stiffness. So stiffness, for example, if you have too much stiffness, let's say like red blood cells, when they have to squeeze through capillaries, the capillaries are really teeny tiny. And red blood cells, if you've ever seen an image of them, like actually trying to squeeze, they have to collapse in on themselves. The only way they can do that is if the membrane is fluid enough and flexible enough to be able to do that. Otherwise, the red blood cell bursts. So DHA and EPA influence the number of these cholesterol domains that are found inside the cell membranes, and not just talking about red blood cells overall. And in this case, we're talking, sure, red blood cells, but we're also talking about endothelial cells, smooth muscle cells, cardiomyocytes, like different heart-related cells. And DHA, if I remember correctly, was the one that allows more cholesterol domains, and EPA may be the one that disallows cholesterol domains. There's an opposite effect there, but both of them ultimately lead to improvements in heart health, at least according to these mechanisms. The EPA, I believe, was the one that most of the reviews focused on, and they also mentioned that it doesn't just affect the cell membrane by being integrated into the cell membrane, but also affects how cells interact with one another, in that cells can produce what are known as eicosanoids, which are produced from other fats inside the cell membrane. So these fats are embedded into the cell membrane. I told you that there were many different molecules found in the cell membrane, which then can be interacted with by phospholipase enzymes to produce arachidonic acid. This arachidonic acid is then acted on by oxygenase family enzymes to turn them into eicosanoids, or eicosanoids, however you want to pronounce it. I'm going to pronounce it eicosanoids because, good gracious, I'm not going to be able to say that over and over again. So eicosanoids are then released and can recruit immune cells. So a little bit like the cytokines, or a lot like the cytokines that we talked about. But in addition to that, they not only recruit immune cells, potentially to be pro-inflammatory, but can also bind receptors on cells like endothelial cells, smooth muscle cells, etc. So in this case, we're talking about endothelial cells and smooth muscle cells, which are found in the blood vessel wall. And in this case, they can bind to a receptor called the thromboxane receptor, which then activates a pathway called the ROCK pathway. And it's spelled exactly like a rock is, ROCK, which then increases the number of focal adhesion proteins. Remember when I mentioned ICAM at one point? So these focal adhesion proteins are proteins that get produced by the cell, then get exocytosed, or get essentially not necessarily released, but get moved from the inside of the cell to the cell membrane and embedded into the cell membrane so that they stick out. They're like, almost think of them like hooks So they stick out of the cell, and then these immune cells can then bind to it, and just like what we talked about with ICAM. Focal adhesion proteins on the cell and can change the endothelial cell morphology as well. So meaning that the endothelial cell and the adhesion proteins, they're also like these different tight junction proteins that are proteins that are bound between cells, as in these tight junction proteins might bind endothelial cells next to each other together to tighten their connection with one another, so that you have less things that are traversing from the lumen, the open space where the blood flow is, to the deeper recesses of the blood vessel. So like the intima, the media that I was talking about earlier when we were talking about intima media thickness. And what this activation of this ROCK pathway does is that it can change the morphology of the endothelial cells so that it squeezes in on itself, which allows greater gaps between the endothelial cells and allows more, for example, ApoB containing lipoproteins to then go into the subendothelium and then potentially accumulate as well as be oxidized. On the other hand, so omega-3s, the way that they act in this is that they can block this process, either directly blocking this process, or they can be embedded into the cell membrane, which actually crowds out some of these other fats that would allow this whole process of eicosanoids to be produced. Another thing that omega-3s do is that they encourage the production of specialized pro-resolvin mediators, which are molecules a little bit opposite of eicosanoids that can also be released from the cell. And when they bind to different receptors on other cells, they can reduce cell migration. They can reduce oxidative stress and reduce cytokine production. So we've already gone over oxidative stress and cytokine production, but why smooth muscle migration would be, or why cell migration would be wanted to be reduced is because in the later stages of atherosclerosis, or like the middle stages of atherosclerosis, which is the plaque buildup and the overall heart disease burden, smooth muscle cells will move from the deeper recesses of the subendothelium and actually migrate, literally move, to other areas that are right under the endothelium and start to create this cap, or try to form this cap over the plaque formation. And that is one of the signs of a progression of atherosclerosis. So the idea being that you can stop this migration and hopefully keep atherosclerosis at earlier stages. I think that there's a little bit of hand waving there. I don't know if that necessarily translates to better outcomes or anything like that, but this is one of the proposed mechanisms at least. Okay, so what about the clinical results? So for this, there have been a lot of meta-analyses, a lot of studies done on this, over 80 studies looking at omega-3 consumption and the effect that it has on cardiovascular disease. Most of the meta-analyses that I looked at show that there is a benefit for cardiovascular disease as measured by myocardial infarction, that's heart attack, as well as MACE, so that's major adverse coronary or cardiovascular events, as well as there was cardiovascular death. So it seems to improve in all those metrics across multiple meta-analyses. However, here's where I have to push back a little bit and tell you why there's a little bit of uncertainty and then bring you back to certainty, is that the Cochrane review, which is one of the gold standards when it comes to meta-analyses, they actually released a meta-analysis as well, including 86 studies, something like that, which I did not include in my analysis, my study analysis, my full study analysis, which is a shame, and I should have done that, and I completely missed the ball on that one. However, I did rectify it in that I did read it, I analyzed it, and I have it included in my upcoming video, which is going to be reassessing the effect on cardiovascular disease. So you're getting a sneak peek on what I'm going to be talking about in that video, or one of the things I'm going to be talking about in that video, in my more like the shortened version of my analysis. So I did read this Cochrane analysis, and they actually indicate that there's no benefit of omega-3s on cardiovascular disease at first glance. However, if you actually read the analysis and analyze it, because they do, I don't know, something in the neighborhood of like 50 different sub-analyses. So it's a ton of work that they've put into this. And it's a fantastic review, but I will say I disagree with how they wrote it in certain respects. Because if you actually look at some of the comparisons that they do, you'll find that actually omega-3 supplementation did improve cardiovascular outcomes for certain metrics that they looked at. So, but then when you look at the paragraphs of them writing, they say it has no effect. Like sometimes they would just say it has no effect, but then you look at the analysis itself, the actual data, and I was very confused by that because it clearly showed an effect. And I'm double checking the references, triple checking the references, and I'm like 99.9% sure that it truly does show an effect. But the way it's written, it just seems so negative, when in reality I probably just would have written it differently. I would have said like there's a small effect or something like that, as opposed to just saying there's no effect, when clearly they show a statistically significant effect. They show that there is an effect, it just happens to be small. So then saying there's no effect is just very confusing to me. Anyway, so it's just like beyond the discussion here. But I ended up looking at those and across multiple sub-analyses. Let's say if you were to just look at straight up cardiovascular mortality or cardiovascular events, in certain sub-analyses there was no effect. So if you're looking at like studies that were under 100 people, or if you're looking at, studies replacing monounsaturated fats with omega-3s or whatever, I'm just like picking random things, then there was no effect in certain sub-analyses. But then in other sub-analyses, especially where they incorporated as many studies as they possibly could, there tended to be a benefit of omega-3s. And that's also true when comparing against other types of fat. So overall, it seemed to me, plus the inclusion of some of these other meta-analyses, it seemed like the evidence was leaning pretty heavily, but maybe not like outright, but pretty heavily in favor of omega-3s being a benefit for cardiovascular disease. Now, a few more things to say on this. One is that there's a little bit of back and forth on if omega-3s may also cause heart arrhythmias. So like an improper beat, especially atrial fibrillation, which is an improper beat in a particular section of the heart, which has been known in the past to be dangerous, mainly because it increases stroke risk. And I will say that none of the analyses indicated a benefit towards stroke risk from omega-3s. So that is kind of notable. I don't know if there's something there. I can safely say that none of them showed an increase in stroke risk. And this is in randomized controlled trials. So these are long-term randomized controlled trials, typically lasting like maybe 2 years, which is kind of the upper end of where you're going to get with RCTs. So it's it's a little bit of a mixed bag when it comes to atrial fibrillation. There seems to be a lot of debate on, from some researchers, they say, yes, it definitely happens. And the meta-analyses that I looked at did indicate that there was an increased risk. I don't remember if the Cochrane review said that there was an increased risk. I don't think that they did, but I'll have to double check that. But the other meta-analyses did indicate that there was an increased risk of this arrhythmia with omega-3 consumption. Now, the idea behind these arrhythmias is that it's specifically the changing of the cell membrane to be more fluid is changing how different ion channels, remember when we talked about chloride, well, there are other ion channels, there are also bigger ones, much more influential ones for sodium and potassium. And those, if they get dysregulated, then that can lead to the cell not activating when it's supposed to. So that's a really general explanation, but that's if you want like actual visuals and stuff, I have a video on the topic. And the cell membrane, when it becomes too fluid, it seems to not allow these channels to function quite correctly. So they don't close when they're supposed to, and they're open when they're not supposed to, which ultimately leads to some cells not contracting when they're supposed to, or cells contracting when they're not supposed to. So that ultimately leads to an arrhythmia. So that's the general idea. But again, like I said, there is some debate on if that's actually the case, if that's actually truly happening. So that's something that we'll have to look into in the future. But currently, the evidence kind of leans in the direction of yes, there does seem to be an effect. But ultimately, other than the fact that it's kind of uncomfortable and maybe a little scary to have your heart doing something a little weird, the overall outcomes are still benefits. So you're still getting protection from cardiovascular disease. You're getting protection from cardiovascular death. So, and you're not seeing an increase, at least by the data that we have currently, there does not seem to be an increase in stroke risk. So, is it just kind of this benign kind of added side effect that's kind of uncomfortable, but doesn't come with the added risk that would normally be attributed to atrial fibrillation and just arrhythmias in general? I don't know. I mean, I'm sure some scientist that actually studies omega-3s and heart rhythms could expound on this to a greater degree, but I don't have the answers to that right now. Okay. The comments on this. So Gorilla Mode asks, would the benefits apply to krill oil? Is there less risk with krill oil, talking about atrial fibrillation, and oxidation contamination than fish oil? Okay, so the oxidation contamination aspect, so contamination is a completely different story. That's like separate molecules or bacteria or whatever gets inside the product. That's something I can't answer. That's just going to come from the manufacturing and good process. But the oxidation, I did look at some studies on oxidation of fish oil, and if it happens, people have way overblown, just massively overblown, based on the current evidence, which isn't much, but like the evidence does not point to oxidation of fish oil being detrimental to your health. I don't know where people have been getting this. It just seems to me like people think, okay, oxidation is bad, so therefore it must be bad that polyunsaturated fats like omega-3 get consumed and then they harm our body. But there's no evidence of that. There are some human trials looking at that, and they have shown either neutral effects or still improved health metrics from eating highly oxidized fish oil or omega-3s. So I see these comments from time to time, and it's just, there's no basis in science on that other than just mechanistic, but that's still clinical data trumps mechanisms when it comes to actual outcomes. And in terms of the other question, would the benefits apply to krill oil? Yeah, I think so. I think the composition of the oil is really what matters. It doesn't really matter the source. If it's krill oil, if it's algae oil, as long as it's DHA and EPA that you're getting, that's what's most important. And if you're part of Physionic Insiders, I went into more detail on like the different types of fats and the amounts and like, what might be the right threshold and all that stuff. It's just there's a lot of nuance that one needs to get into. And I think some of it, as I mentioned with like atrial fibrillation, is stuff that we just haven't quite figured out. Although, like I said, there have been a lot of studies on omega-3s. And one more thing that people ask about: fish oil. Do you need to supplement with fish oil? No, you don't. You can absolutely eat fish. That's another option. Maybe if you're eating fish, you can maybe cut down on the supplementation with fish oil. You probably don't need to be supplementing with fish oil or krill oil or algae oil, whatever, and eating fish at the same time. You can probably just do one or the other. Rob Smith, how does the consumption of fatty acid C15 affect this process? So I'm actually going to be covering C15 in just a little bit. So stay tuned for that. But to answer the question, it does not affect the process, at least this is educated opinion on my end. And yeah, I would just say it doesn't have an effect, mainly because the evidence around C15 is, well, let's wait till later. All right, creatine on bone and muscle in older individuals. Well, the reality was I looked at a meta-analysis on bone, and I looked at a meta-analysis in muscle. And on bone, the researchers indicated that there was no effect of creatine on bone. Now, before I keep going, I'd like to point out, because people were then saying, well, we won't see any benefit of creatine without resistance training. And both groups were resistance training. The control group or the placebo group and the creatine group were resistance training, and there was no effect seen. However, there were some big critiques that I had of the analysis, not necessarily the analysis itself, but like, should it have even been done? Mainly because half of the studies, there were only like 6 studies, 5, 6, 7 studies, something like that, involved in the analysis, and only 6 of the 6 studies, 50% of them only lasted maybe 3 months, which for bone is entirely too short of a timetable to assess any sort of meaningful differences, unless the effect is massive and in very quick form, which it is not with creatine. It is maybe in muscle, but not in bone. So my conclusion is not that creatine is ineffective for bone in older individuals, but that we simply don't have enough data, quality data, to really assess if creatine is a benefit to bone. So for the time being, I would say inconclusive. We just have to wait for more research. In terms of muscle, obviously we've got plenty of research saying that creatine is beneficial for muscle in younger individuals, but I wanted to know if it also happened in older individuals. And the answer was a resounding yes. So creatine was still beneficial in increasing the lean mass, which obviously could be different tissues, but likely it's dominant in muscle. The second thing that it helped with was muscle strength as well. So it improved muscle strength across all the metrics that were seen. And then I went into some more specifics for the insiders with like the frequency of dosing, the effect that it has on lower body strength versus upper body strength, and things like that. But the overall point was that creatine does seem to benefit muscle strength, muscle size likely, and it's inconclusive on bone. So still a fantastic supplement to continue to consider using if you want. Okay, comments. Vigor2. People hate hearing it, but the best thing for the last 1/3 of your life is to have cared for it in the first 2/3 of your life. You'll have more in the tank heading into your later years. Well, I certainly don't hate hearing it. It's absolutely true. The sooner you can get onto physical exercise, nutrition, all that stuff, the better your health will be. But the next best time is today. So any sort of positive change towards that can have tremendous effects for later in life when you're in your 70s, 80s, 90s, and beyond. Mike Levinson, 70 years old here. I lift 3 times a week, 1 day cardio. I take approximately 5 to 6 grams a day of creatine. It's not a miracle supplement, but it does make a difference in building muscle for me. Yeah, which is consistent with what a lot of other people have reported, and obviously what many, many, many studies have reported as well. So thanks for sharing, Mike. Okay, we're in the home stretch. Last 3 or so topics left. I'd like to cover a study on age-reversing antibody therapy. So I thought this came on my radar. I don't actually know how, but I thought it was fascinating. So I decided to do something for myself and just read it and go into a deep dive on it. So the way this study worked was that they were trying to use immunotherapy essentially. So they're trying to replenish the immune system of old mice. And they're already going to be, I think, moving on to humans for the next stages for this. And there's some really promising research and promising ideas related to this. So immunotherapy is essentially just using your immune system to take care of different problems. And as we get older, what happens is that we get an imbalance of immune cells. So we get more myeloid immune cells than we do lymphoid immune cells. The difference between those two is that myeloid immune cells are kind of like a first line of defense. Think of like macrophages, neutrophils, things like that. So these cells scavenge around. They're just kind of like patrolmen. They're just kind of walking around looking for burglars. And once they encounter like a bacterium or whatever, then they end up eliminating it. Just kill it. But on the other hand, if something becomes too overwhelming or they just can't handle it, there's an exchange of responsibilities essentially. Not that myeloid just like give up and just sit back, but they essentially recruit this other system. This other system is the lymphoid system, and it's far more specialized. So the myeloid system and these kind of intermediate systems between the two will present antigens or parts of the pathogen that they're trying to get rid of, that they're having trouble with. And that will then activate the lymphoid system to then generate many different T cells and many different B cells and many different immune cells that will ultimately be very targeted on exactly that pathogen. So they're not as effective for general like Sentry, but they are very targeted at attacking that one pathogen that they've now been trained to attack. So your immune system is an incredible thing. However, as we age, we have more of these myeloid cells and we have fewer of the lymphoid cells. So obviously that means that if you have a dangerous infection, think about it, or if you have cancer, or if you have any sort of condition that has any relevance to your immune system, then you don't have the requisite lymphoid cells there to deal with whatever problem is there. And that could then lead to people dying much earlier than they normally would. So what this research group did is they tried to find a way to get the stem cells that are found in the bone marrow to be more balanced. So we have these stem cells. The way that all of our immune cells are produced is that these stem cells, called hematopoietic stem cells, will differentiate, will turn into the different types of immune cells. These immune cells, or at least like these progenitor cells, these kind of beginning stages of these immune cells, will then be more myeloid, or they'll be more lymphoid, and then they'll continue down that path to become whatever myeloid or lymphoid, a mature version of that. But the researchers are trying to address specifically the stem cells, not the later stages of myeloid or lymphoid development, but specifically the stem cells that may be more biased, or this old phenotype, quote unquote, old phenotype, that would be more biased towards creating myeloid cells. So the stem cells would divide, and then they would turn into myeloid cells, as opposed to turning into a balance of, maybe some myeloid, maybe some lymphoid, like they do in healthy young individuals. So the way they addressed this is that they had to figure out a way, how do we tell the difference between the balanced stem cells that we want, the ones that create both myeloid and lymphoid, and the myeloid biased stem cells? How do we tell the difference? Because they're both stem cells, they're both hematopoietic stem cells, so how do we tell the difference? Well, they were able to characterize that the myeloid stem cells have high levels of a protein called CD150, and the balanced hematopoietic stem cells have low levels of the CD150 on the surface of the cell. So now we have a distinguishing feature to separate out the two groups. On top of that, they did some further work where they figured out some other proteins as well that then are able to target those specific proteins. And the way they're able to target that is by producing these antibodies. These antibodies then bind to the proteins that are more highly expressed in the myeloid biased stem cells, and essentially encourage them, or have the immune system come in and destroy those myeloid immune cells. It's either the immune system, or they can also undergo apoptosis, which is programmed cell death, because the researchers also blocked a protein, CD48 or something like that, CD63, I can't remember. But they blocked a protein that would normally block apoptosis from occurring. So they blocked the blocker of apoptosis, which then allowed apoptosis to occur more readily. So now they're diminishing the stem cell pool of these myeloid biased stem cells. And then the question was, well, are we getting more of these lymphoid stem cells? And the answer was yes. They ended up with this antibody cocktail. They were able to increase the number of these balanced stem cells. In addition, they did a few more experiments where they did vaccinations of the animals to see if the animals would be able to react to the vaccine, which is obviously really important when you're talking about older individuals. Older individuals, when they're getting vaccinated, it might not be as effective. Well, the reason is because, or at least one of the major reasons is this. So if they can fix this, then vaccinations should be much more effective, meaning that with the vaccine, you would see, because you're applying like maybe a particular section of the pathogen that you're trying to vaccinate against, now you're stimulating that lymphoid system to generate cells for that pathogen. So, and what they found is that, yes, they were able to increase the number of these lymphoid cells. They also had some experiments on spleen, which spleen is important in immune function as well. But ultimately, the point being that you see increases in these cells. The study wasn't perfect in certain respects. I wish that they had comparisons against, well, I suppose I don't need to get into it for the podcast. The point being that the study wasn't perfect, but it was very, very interesting. And I think also that this would apply not just to just in general increasing the number of lymphoid cells, but that would actually translate, if the effect size is great enough, which is something that's definitely needs to be figured out. But if the effect size is great enough, then it could affect cancer rates. Like you could be able to fight cancer much, much more potently. Because remember, cancer tends to show up much later in life. And one of the reasons is because of the lymphoid system not being able to handle new cases of cancer. So it's just one of the mechanisms. There's certainly more. So it'd be a huge anti-cancer treatment, not necessarily as like an immediate, like, oh, I have cancer, so therefore I'm going to take it, but as a preventative, actually. And what was really cool is that they only applied this once, and it lasted months. The benefit lasted months. So think about that. What if this applied to humans? Of course, that's a what if, but what if this applied to humans and a person just takes one pill or it'd probably be a shot if I had to guess, one shot of this preventative cocktail, and then it lasts 6 months or it lasts a year, that your entire stem cell system kind of regenerates so that it becomes more in a youthful state. Now, one question that people had was, well, I don't understand. If we're eliminating stem cells, these biased stem cells, how is that going to improve the stem cell population? Stem cells renew themselves. So, and our stem cell pool decreases mainly because we start to accrue a bunch of damage. Those stem cells essentially just become defunct. They become senescent, or they just become less able to replicate themselves, whatever it might be. So our stem cell pool decreases over time. And what this would do is it would preemptively eliminate the ones that might be crowding out some of the younger, more functional stem cells, and allow those stem cells to then refill the pool. That's the theoretical idea. I don't know if that's exactly how it would work, but that's my understanding of how it would work. Okay, so anyway, I thought that was really cool, and I thought I'd share some comments on this. Azzy Freeman said, maybe they will do a study to see how this affects people with autoimmune issues. Okay, yeah, so that's a great point. That's another one where maybe, especially if a person has an autoimmune issue that occurs later in life, that if you can target the proteins that would be expressed in the stem cells that are creating these problematic immune cells, because obviously the profile would be different. You'd have to figure out what that profile is, what the proteins are that are specific to those stem cells. Then you might be able to eliminate those and then be able to cure a person with autoimmune, or at least treat them so well that they feel like they're cured. They just have to get a shot every 6 months or whatever. Anatoly Dyatlov, that is revolutionary. I agree. I'm already hyped to get therapy like this and hope it will become available in the next few decades. Actually, I'm not that optimistic usually on most things, but I think on this I am optimistic. Immunotherapy has been around for a little while. I remember when I was first getting into my PhD at the University of Florida, I did some interviews there for different PhD programs, and they had a bunch of researchers working on immunotherapy, and I'm sure it's being worked on many, many different places. And so that would not at all surprise me. I think that I'm more optimistic. I don't think it's going to take decades. Maybe a decade. I don't know. But I think we're going to see some really great advancements in the next 5 to 10 years. I don't think it's going to take decades for us to get to a point where we have at least clinical trials and hopefully some kind of preliminary treatments. I don't know how the safety assessment of that is going to go, because you are messing with your stem cells, which is a major thing that you have to be very careful not to mess anything up. But I'm a little more bullish on this than I am many other therapies and supplements and whatnot that have been proposed and put out there. All right, second to last topic is the 2 age-related issues. I did mention that over the age of 60, these 2 issues pop up. So I'm going to keep this relatively short. The 2 things are a drop in metabolism, which people seem to think that your metabolism drops around age 30. That is very far from the truth. Your metabolism actually remains incredibly stable for the majority of your life. It's not until you're about 60 that you start to see a drop, and even then it's not huge. And then it becomes a little bit more noticeable around age 70 and beyond. But while the overall effect is not solely muscle-based, one part of it is, and it's a large part of it. So it's not completely because of your loss of muscle, but it is a large part. So the solution, of course, as you can guess, is to weight lift and to make sure that you're resistance training to a point that you can maintain your musculature as best that you can. The other factor is something that almost no one ever talks about, but was talked about in my courses and in some of the labs that I did some work in, and that's a drop in power output. So power is the speed at which you can move a particular weight in very simplistic terms. So while strength and muscle are important and critical, I would say, for overall health, especially after the age of 60, what we don't realize is that our power output is also something that needs to be considered. So doing exercises or doing something that's maybe very light on weight, that's not actually very heavy for you, but you move through it very quickly, through that range of motion very quickly, can improve your power output. Actually, powerlifters do this all the time, where they're not necessarily just working in the really heavy ranges. Sometimes they'll back off and do work at much lighter ranges, but try to lift the weight as quickly as possible. So those 2 things, power, which is something that decreases very quickly at around middle age. Actually, it occurs usually before 60, but around middle age, it starts to occur. And it's very important because when you're, let's say, walking and you trip and you fall, especially older individuals can break something or, completely mangle their body, which leads to hospitalization, which is one of the number one leaders or relationships to death. Obviously, it seems almost obvious that, oh, wow, Nick, you're saying hospitalization has a relationship to death? Shocking. Well, I mean, it's obvious, yes, but specifically that type of hospitalization, I should say, is highly related to ultimately leading to mortality in just a very short period of time, because everything just kind of goes downhill once you're immobile and you're unable to do anything from that point on. We're talking, when you're in your 60s, but especially in your 70s and 80s and beyond. So power will allow you to trip and catch yourself before you have this disastrous consequence. So power is not something that people talk about, and it's not something that's obvious when you just look at someone, but it is something that's insidiously decreasing over time, and just weight training is not necessarily going to be enough. You have to implement some power specific training as well from time to time. So anyway, just some things to consider. Some comments here. Grayley said, a caution from a 54-year-old that had returned to resistance training after a 7-year break. Doing explosive lifts when you're older can get you tendinopathy, which is like hurting tendons. We'll just put it that way. Very simple terms. Rotator cuff, biceps, patellar, piriformis. I've messed up all the tendons in the last 8 months. The risk is probably higher if you're not an experienced lifter, since in the beginning, strength and force output develops faster than tendons. So after 1 year of lifting, you're just strong enough to hurt yourself. Yeah, that's all a good point. That was actually one of the pushbacks I got on that video, that people were like, well, if you're older, you probably you shouldn't be doing explosive movements and whatnot. I think that people are failing to realize that you can do explosive movements you're not supposed to do explosive movements at the same intensity that you would do your normal movements. You're drastically dropping the intensity so that you can move through the range of motion without affecting these other systems like the tendons, your ligaments, and all that stuff. And also, you're not doing it necessarily every single workout. You can do it every workout, maybe just like one set every workout, or you can do just have like one dedicated workout, or you can just have one workout where you do it. Of course, everything is going to have to be changed based on what the individual can handle. But also, when you're talking about percentages, when it comes to like weightlifting percentages and stuff, you're simply not doing this kind of explosive stuff. I mean, maybe if you're younger, you can, but you're simply not doing this explosive stuff at like 80% of your one repetition maximum or 90% of your one repetition maximum. You're going drastically lower than that just to go through those motions. And especially, I mean, sure, for older individuals, you might even have to go even lower than that. But it's either that or you continue to lose your power with no way of fixing it, which, I mean, granted, I don't have personal experience with this, so maybe I'm just completely naive. But then again, I also think to myself, if you're doing like 30% of one repetition maximum, that is very, very light. So if your tendons are that weak, I think then I would probably put it this way. If you have real trouble, like where you're experiencing a lot of tendinopathy and whatnot, you should probably focus on strength training alone and not worry about any sort of explosive movements, and just kind of build up those tendon strengths. And then after a certain time, let's say 6 months, once you feel more robust in like your ability to do to lift weights and whatnot, then you can try to do some more of this explosive stuff, still at a drastically cut percentage. So then the relative amount is at this relative amount that's much lower, I'm just throwing out a number, 30% or whatever it might be, and your tendons have been under much higher load. So once you're doing your much lighter load, even if it is explosive, your tendons should be able to handle it. At least that's how I would approach it if I was having continuous problems with my tendons. And of course, you need to be changing things based off of your personal circumstance. But hey, who knows? Maybe I'm just naive, which is entirely possible. But regardless, one way or another, that's the solution that you have to do. You have to do explosive movements. There's no, as far as I'm aware, there's no way to go to do it differently to increase power output, because the power is specifically hinges on speed. So if you're not implementing a speed component, then you're not going to be improving your power output. I mean, I'm sure there's some parallel between like a strength. As your strength increases, your power might increase a little bit, but it's just not going to be nearly as direct of an effect. But hey, maybe it's something I can look into in more depth, especially on channels and brands that specifically focus on exercise and focus on muscle. See, these are the kinds of things that, when you go to a lot of like weightlifting channels and bodybuilding channels and stuff like that, which I've gone to so many of them in the past because I was really into it and still am, but just a little bit less. There's always this complaint that they talk about the same thing over and over and over again. And I think that's generally true. There are some channels that try to mix it up a little bit, but there's a wealth of information out there that people could be getting into that they're just not. And this is a great example of like how this is a fantastic problem to try to fix for people. Well, I'm 75 years old. I can't do explosive movements. So how would I improve my power output? That would be a great question for somebody in the exercise, like specifically in the exercise space. I mean, I could look into it myself because I do have a master's in exercise physiology, and certainly I could look into the research. But it's something I think maybe someone with more hands-on knowledge, this maybe even somebody who's in their 60s or 70s that has a bunch of this background knowledge could address. Anyway, let me move on. Seattle Gurley said, in my mid-40s, I was seeing a decrease in muscle strength. Not extreme, just getting harder to lift a big box of cat litter out of the shopping cart. Back into the gym I went. Now I'm trying to get all my friends to lift weights. Yeah, so it's sometimes the small stuff. You just realize, wait a second, this used to be a lot easier for me, and now it's become a lot more difficult. And I mean, kudos to you, seriously. And also to Grayley earlier, just going back to the gym at 54, Seattle Gurley going back to the gym just from noticing that small thing. It's like you have that shift of like, I need to do something about this. And a lot of people would just let it go, like, oh, I'm just getting older, I'm just getting weaker. But that mentality is so commendable to say, I'm going to fix this. And there's a lot you can do, really awesome. Okay, let's round this out. The final topic, Fatty 15, the company, or C15, which is a odd chain saturated fat. So if you pull all the carbons together in a chain, you're going to have 15 of them. You have other odd chain fatty acids or fat molecules like C17. And this has become all the rage. There's been a ton of hype. I don't know if it's just me. I've seen a ton of marketing for this supplement called Fatty 15. They have a TEDx talk or TED talk, whatever the difference is between those. And I've seen all over Instagram and YouTube and shockingly, it's crazy to me how many people have created videos on C15 and said it's a good product. I'm talking about like science channels. I'm talking about longevity channels. I'm talking about channels that are supposed to be evidence-based, supposedly, and they're talking about C15 like it's the next frontier of health. And I cannot express to you how much I disagree. I read 8 studies to get to the bottom of it, and I didn't know anything about C15. I read a bunch of studies, went to the Fatty 15 website, and the Fatty 15 website makes a ton of claims about, by comparison to like omega-3s, they say that it's like it strengthens the cell membrane. It's just like that's marketing talk. What does that mean? Strengthen the cell membrane. It is better than omega-3s because it repairs 2.5 times or 3.5 times more cell types. It's like, but then you look at the research that they cite for that, and it's research that doesn't have proper statistical comparisons, doesn't have proper statistics as a whole. The evidence that they cite actually doesn't show that. It has nothing to do with cellular repair. It's using a cell-based system, which they claim is supposed to be representative or close to representative of clinical outcomes. It's not. It absolutely is not. It's just nonsense. I mean, the whole thing is just crazy hyped up. And there's 2 clinical trials on it. One of them is partly funded or partly in relationship with the Fatty 15 company. And that study found some small benefits on liver health. So that's good. I think the study is pretty well done, to be honest. But the effects were on liver health and basically nothing else. There were some slight effects on like LDL cholesterol. The magnitude of the effect is minuscule. Like you could sneeze and there'd be an effect. It's that small. The other clinical trial has a few issues with it, but it is not associated to the C15 selling company. And that trial showed no benefits of C15. So and then some of the other studies, there's one correlation study. They do have some adjustments, like some statistical adjustments that strengthen the case for a correlation study. But it's really difficult to do statistical adjustments on a single molecule. It's a different story when you're talking about did physical activity, which is like an all-encompassing, it does many things. Or if you're doing statistical adjustment for diabetes, that affects many different organ systems. But to look at a single molecule when there are thousands of molecules and then saying, well, we adjusted for weight, caloric intake, which are important adjustments that it's good to do. But even if you adjust for all those things, you also have to adjust for thousands of molecules, which you can't do, but you can adjust for physical activity because it's an actual overall outcome. It's an overall thing that you can do, as opposed to C15 is just a single teeny tiny molecule in a sea of other molecules. So adjustments are kind of they're I don't want to say useless, but it seemed to me close to useless. Anyway, the point is that study found that there was a good relationship, as in a negative relationship with diabetes. So if serum or blood C15 levels were higher, incidence of diabetes was lower. So that's good. But to me, that's for other people that constantly claim correlation, is not causation. This is a prime example of when you could easily say that, because usually if there are adjustments, the proper adjustments that are made in what's called a multivariate analysis, then it's not that you can say that correlation is causation, but you're clamping down and you're saying, okay, well, we're controlling for all these other variables. So it's likely this, very likely this, even if there's still a little bit of a shadow of a doubt. But with this, there's tons of doubt that's still available. But let's roll. I mean, what is undeniable is that there is a relationship between higher blood values of C15 and lower diabetes risk. What is deniable is that a cause and effect relationship? That's my question. And I just don't really believe that, at least based off of the evidence that I've seen so far, which is mainly based on cell studies. Let's see what else. Yeah, the omega-3 comparison, it was horrible. Just such a bad comparison. It didn't tell you anything. And then, one more thing. They said omega-3s were cytotoxic, so they would kill off 4, I think, or a bunch of cell types. I don't remember the exact number, but it would kill off a bunch of cells at a high concentration, while C15 did not kill off any cells at a high concentration. But then I looked at that study that they cite, And they show no evidence, zero evidence of that. As in, I went into the supplemental material as well to look at their data, zero evidence of that. It's like they didn't even do the experiments. I'm going to give them the benefit of the doubt and assume that they did do the experiments, but they just don't provide any data for that. They say it, the researchers say it, which the researchers, by the way, are the same people that wrote the article for C15, because it's the company, one of the co-founders is the researcher, and the other co-founder is the one who wrote the article comparing omega-3 versus C15. So the researchers, aka the co-founder, says that this in the research paper, but then doesn't provide any evidence for that. So it's like that kind of stuff. What else was there? There's some claim about ferroptosis. Ferroptosis, I've talked about apoptosis briefly. Apoptosis is programmed cell death. There's a relatively new type of cell death called ferroptosis. There's another one called necrosis, but we'll focus. There's this new review on ferroptosis in C15. I briefly skimmed it, 100% honest. I have not read the whole thing. I skimmed the whole thing, but I just haven't done a deep dive into all the studies and whatnot that they link. And the review mentions that the relationship between the mechanism by which C15 improves health is by, or one of the mechanisms at least, is by reducing ferroptosis. Ferroptosis is a type of cell death that occurs at the mitochondrion. You have a what's known as an antiporter, which allows a certain molecule or ion into the cell, and then another ion, it kicks out of the cell, or I guess it's not an ion in this situation, we're talking about a molecule. It's glutamate that it kicks out of the cell, and I believe cysteine that gets imported into the cell. And the cysteine then gets used to create glutathione, which then gets used by glutathione, I think peroxidase, don't quote me on that exact name, to eliminate reactive oxygen species and eliminate these different components of damaging molecules. Now, apparently there's some sort of cell fragility syndrome, which I am 100% unconvinced actually exists so far. That may change in the future, but from the little bit of reading I've done on it, it just seems like another just buzzword that's just being thrown out there. So cell fragility syndrome occurs when you have this lack of intake of cysteine into the cell, and then you don't have the production of glutathione, and therefore the cell and the mitochondria ends up essentially like dying off. And then you have ferroptosis. You have the cell death pathway gets activated, and then the cell ends up dying. There's also an iron component where iron is involved in the process as well. Anyway, C15 is proposed to lower the amounts of this like cell death, this particular type of cell death. Now, I did look at those few sentences where they tried to make the link between C15 and ferroptosis and stopping ferroptosis, and there is basically no evidence. First off, they make the claim, and they say there's all this evidence, but then they provide zero citations next to that sentence. I'm like, all right, great. So then you read further into the review, and you get to like a paragraph or two where they talk about it, and they have 3 references. One of them is a paper in mice. Don't have a problem with that, but it's in mice, and they actually fed inulin, a fiber, to the mice, and the mice's microbiota created a C15, and then that led to some improvements in some gene signatures, or some changes in some gene signatures, which are associated with better health outcomes. That is a lot of hoops to jump through to get to the idea that C15 is ultimately leading to better health outcomes, like reduce diabetes, reduce obesity, all these different health outcomes, reduce cardiovascular disease. That is a lot of hoops, especially on a mouse study. To be fair, this mouse study did also apply C15 to the mice directly, and they did find these effects. However, none of that is related to ferroptosis. None of it. I even checked the paper. I did like a search for ferroptosis and apoptosis, and those words weren't even mentioned in the study, and yet it's being referenced as the reference, or one of the few references for how C15 is affecting ferroptosis. Okay, great. Then the other study that they reference is their own study where they looked at mitochondria, because they're trying to create the relationship between C15 and improved mitochondrial health. And they use a single mitochondrial measure in a immortalized cancer cell line, which, to be fair, that's perfectly fine, but that is literally the bottom of the barrel level advice, or not advice, but evidence. The bottom of the barrel. I don't have a problem with it. It's just you have to go up the rungs and continue to prove that that's the case in other cell types. Then you have to go onto tissues. Then you have to go onto animal models. And then at animal models at that point, you can probably start to make this claim. And then you should ideally try to go up to other models, which obviously are more difficult, like human models. Okay, so they cite their own paper where they have a single experiment where they applied C15 to these cells, and the cells' mitochondria ended up generating less reactive oxygen species, except the amount of difference is like 10% or 15%. And the control has multiple problems with it, which I don't need to get into, because I'm just going to go off on a tangent, which I basically already am. And they also misspelled the word mitochondria on the data itself, which, I mean, isn't a huge deal, and you could accuse me of nitpicking, but then I'm thinking to myself, like, if this is your only piece of data, how have you not looked at it to make sure that at least the spelling is correct? It just gives off this sense of like, we don't really care. We just want to throw it out there. They also report relative changes only, which isn't the biggest deal, but it kind of feeds into this idea of like, okay, we're giving you the least amount of information so that we can make this claim, and then we're just going to like shuttle it away. So they reference this particular study, and I mean, I won't deny it reduced reactive oxygen species, but I'm thinking to myself, I imagine there are many other molecules that do this to a far greater degree than 10 to 15%. What else? Oh, then they referenced a dolphin study. That was their three pieces of evidence for ferroptosis as far as I could see. So none of those directly measure ferroptosis. Yet in the title, if I remember correctly, in the title, it says ferroptosis. So they're making all these links, but the links are these like hoops that the reader would have to go through to maybe get to the conclusion that it could have an effect. And it's just such bad science. It's so bad. So let me get to my final point. Hype outpacing the science. I am frankly appalled by the number of people that are on social media. You type in C15 at the time of this recording and look at the number of people that have talked about C15. They've even flashed the study titles up on the screen and whatnot. And yet the science is so bad and no one mentions anything about this stuff. Why? Are they just not reading the studies? Are they just like incapable of realizing that these studies are just absolute crap? I don't get it. I really just don't get it. Anyway, that's my strong opinion on C15. To be 100% clear, I'm not saying C15 can't have evidence behind it, and maybe it does have a benefit to our health, but the amount of hype and the amount of like, oh yeah, we have so much science behind this product, the amount of hype that's been built up around this thing, and when you look at 8, 9 studies on the topic, and they're all just such bad studies for the most part, it's really difficult to imagine how did they get to this level of excitement about this product. And the amount of marketing from people that, like, I even saw somebody market, a registered dietitian marketing on Instagram with Fatty 15. I'm thinking, have you no shame? Have you not looked at these studies? This is horrible. Okay, I'll stop. I promise I'll stop. I get passionate about this stuff because it's just insane to me. Anyway, my opinion currently, it may change if maybe 5 more studies come out and they are like really well done and they show C15 clearly has tremendous benefit. I will absolutely say C15 now has the evidence behind it, but currently the C15 does not have the evidence to support it. And I would say, save your money. Do not buy C15. Okay, last comments. Danny, dude, thank you so much. You're welcome, as always. The company is charging $100, wow, or more for their product. I actually didn't know how much they were charging. Save me some money. Yeah, you're very welcome. Definitely save those $100. Somerset girl, I fell for the C15 hype, bought a 90-day supply and took them consistently. I didn't notice any of the supposed benefits, so haven't bought any more. One factor has occurred to me. I consume quite a lot of full-fat dairy in the form of kefir and cheese, so it's possible that I was already replete in this nutrient, so taking extra was not going to have any benefit or effect. Yeah, that's absolutely that's absolutely possible. One benefit of the studies is that they did separate out those that achieved a higher level of C15 in the blood, and then they eliminated the participants that did not see an increase in C15. And to their credit, they did see the liver improved in only the people that reached the higher C15 values. But to temper that, just to remind you, the effect was pretty small. It's not a massive effect. It's not like your liver's suddenly going to regenerate or anything like that. It's a small effect. It is there, but it's a small effect. So it's possible, definitely. Anyway, those are my opinions on C15. The final consideration here is housekeeping. I already spoiled this, but I finished my PhD back at the end of June. So I'm officially done with that. I feel incredibly relieved and a little shell-shocked. And that leads me to, what am I going to do now? So I love Physionic. I love what I'm doing. I get incredibly passionate about it, and I can't seem to stop thinking about just reading research and putting out videos and just like having a go at this. So that's been my plan. That's what I'm doing. I'm just going to keep creating videos, hopefully continue to build a business out of this. And hopefully there'll be enough people that want to support my work over time that I can continue to do this in perpetuity, because I would really love to be able to just keep at this. I don't think I ever want this to be like a massive endeavor where I have a whole like teams of people helping me with all this stuff and all these different, it's just overwhelming. I just like to keep it just me. But the other thing that I'm considering is potentially doing some teaching, but I don't want that to take away from Physionic if I can. Just like financially, I just have to figure things out. in terms of like what's feasible and what's not feasible. And time is also a massive resource that I don't have a whole lot of time. I don't have a lot of, oh, you don't have a lot of time. Yeah, wow, shocking. So there's a lot of considerations, but I'm really excited about the future. I'm really excited about where Physionic is going, and I'm going to continue to put my best foot forward. And lastly, on my notes, I always want to make sure that I'm thanking everyone for getting to these points. I mean, I'm not even talking about just specifically in the podcast. Yeah, the podcast ends up being super, super long. What is this one? Over like around 4 hours long. But just in general, it's always incredibly humbling to have people that frankly harass me for my opinion on like C15, for example, which, by the way, those videos that I did on C15 just flopped. I mean, I got so many requests, like the most requests I've ever gotten. People were getting angry at me that I hadn't released my investigation on C15. And then it releases and the videos do really badly. It's just comical to me how some people can feel so passionate about like learning about something, and then the greater majority of individuals just don't care. So anyway, it was still fun for me to look into C15, and it is humbling that people actually care about what I have to say on the topic. I may have gotten a PhD now in this kind of stuff, but still, even beyond that, it's an incredibly humbling experience to be able to sit here and discuss science and try to put your best foot forward and try to make a difference in the world. So, yeah, thank you. I really can't express that enough. And that's always going to be the case. Anyway, this is where I'll end the quarterly podcast, if you want to call it that, or every 3 to 4 or 5 month podcast. And thanks again for listening in. If you got to this point in the podcast, seriously, it's like shocking when people do. But I'm ever grateful. And until next time, have a good one. See ya.